Hello everyone and welcome to another warm-up. Let's get going. Which muscles and nerves are derived from the first branchial arch? So the first arch is the M's and the T's, which makes it a little bit easier to remember that the muscles of mastication and the mylohyoid muscle comes from that first arch. Also with the T here, remember the tensor tympani and the tensor villi palatini. And then for the nerves, again, remember the M's and T's for the maxillary and mandibular division of the trigeminal nerve. Next question. Which amino acids are found in large concentrations in elastin? So in elastin, you're going to find glycine and proline. Next, what is the mechanism of action of adenosine as an antiarrhythmic? So adenosine increases the outward potassium current, causing hyperpolarization of cells and decreasing intracellular calcium, so it basically stops the heart transiently. All right, that's it for the warm-up. Let's get to that lecture now.
This is the first video in the immunology section, and I'm sure some of you are dreading this. But immunology is very, very high yield for step 1. Immunology is very detail-oriented, so there are a lot of little things they can ask about, and it can be difficult for students to learn these details. So don't overlook immunology. I'm going to try to focus on just the details you need to know for step 1. You really don't need to go outside of these lectures and your step 1 review book to know your immunology for step 1. We're going to cover everything that's high yield. So in this first immunology video, we're going to talk about the basics of the immune system and some anatomy and some histology and some conceptual stuff. There are two big categories of organs that can make up your immune system: primary lymphoid organs and secondary lymphoid organs. The primary lymphoid organs are the bone marrow and the thymus. Now, you may not think of your bone marrow as an immune system organ, but that's where all of the cells in the immune system come from: T cells, B cells, and macrophages, and then they have to travel to other sites and mature. For example, the T lymphocytes mature in the thymus, which, again, is another one of the primary lymphoid organs. But that's T as in T lymphocyte, or T cell, and that's also for thymus. Then, the secondary lymphoid organs are just as important as the primary lymphoid organs. These include the spleen, the lymph nodes, and the mucosa-associated lymphoid tissue, or MALT. We're going to cover the spleen in another video, but let's take a look at the lymph nodes. Lots of immune cells hang in the lymph nodes until they are needed. Unlike the GI system or the respiratory system, which are confined to very specific parts of the body, the immune system has to be everywhere, so that whenever there's a breach of the body's defenses, immune cells can get there in a hurry. So lymph nodes are found throughout the body, but they tend to be concentrated in areas that are near openings to the outside world, such as the neck, to be close to the mouth and the nose. About half of the 600 lymph nodes found in your head and in your neck. There are also lymph nodes clustered in the mediastinum, because obviously things get into the lower respiratory tract and the lungs. There are inguinal lymph nodes clustered around the groin, because it's close to the genitals. And there are lymph nodes in the axilla, which drain the breast, because the nipple is a potential entry point for pathogens. In general, the histology of lymph nodes is not terribly high yield for step 1, but I do want to talk about it for a moment, because there are a few important points, and it helps us to understand the interconnectedness of the immune cells. It's fair game for step 1, but I don't want you to get too bogged down in these. So, what exactly do lymph nodes do? Well, macrophages hang out in the lymph nodes and filter and clean the lymphatic fluid, which is essentially the acellular part of the blood, the serum. So macrophages are essentially filtering and cleaning the blood. Lymph nodes also house lots of lymphocytes, B cells and T cells. This is the site of B and T cell activation, proliferation, and differentiation. And it's the important site where the plasma cells synthesize antibodies that are going to enter our circulation. One thing that's worth knowing is where you might find these particular cell types within the lymph node. This is a drawing of a lymph node. The green vessels coming in are the lymphatic vessels, the afferent lymphatics. Lymph nodes usually have lots of afferents. This is where lymph is collected. And the blue one is the efferent lymphatic, and that's where the collected lymph is exiting the node. Then the node itself is this multicolored thing. The outer zone of the lymph node is the cortex, which we've got colored light blue. The innermost part is the medulla, which is dark blue. And then the zone sandwiched between the cortex and the medulla is called the pericortex. And the cortex, you have areas called follicles, which are just regions where there are lots of lymphocytes packed tightly together, specifically B lymphocytes. This is the site of B cell localization and proliferation. So where do you find T cells in the lymph node? Right next to the follicles in the pericortex. The pericortex also contains venules, which is how B cells and T cells get to the lymph node. And then another very important point is that if there's a viral infection and your body is proliferating T cells in order to attack the virus-infected cells, the pericortex is going to become enlarged. In other words, during a massive cellular immune response, the pericortex becomes enlarged. It makes sense, right? If you have a virus infection, you need to have the T cells to counteract the virus, and that area houses the T cells, which is the pericortex, so it needs to expand. In contrast, the pericortex is not well developed in patients with DeGeorge syndrome because you don't have mature T cells in DeGeorge syndrome as you don't have a thymus to mature them. So patients with DeGeorge are not going to have highly developed pericortex. Going back to our drawing, what kinds of cells do we find in the medulla of the lymph nodes? The medulla has little stacks or cords of cells called medullary cords. These cords are made up of B cells and also plasma cells, which is what I want you to remember. The plasma cells are fully mature, fully differentiated B cells that have just become little antibody factories. So plasma cells in the medullary cords. And the spaces between the cords are called the medullary sinuses, which is where you find the macrophages. So remember, M for medulla and M for macrophages. Another type of secondary lymphoid tissue I want to mention is mucosa-associated lymphoid tissue, or MALT. This is lymphoid tissue that is found in the mucosa of the GI tract, the respiratory tract, and so on. For example, in the lamina propria of the intestines, there are numerous lymphoid follicles, which are sometimes called GALT, or gut-associated lymphoid tissue. Up to 70% of your immune system tissue lies in that GI tract. One type of GALT is found exclusively in the ileum. These are called Peyer's patches. Peyer's patches are unencapsulated lymphoid tissue found mainly in the lamina propria of the ileum and also extending into the submucosa. So this unencapsulated lymphoid tissue is not like lymph nodes that have a capsule surrounding them. And these Peyer patches are only found in the ileum, and that's how you can tell the ileum from the jejunum, microscopically. Here's an image of a Peyer's patch. You can see all of these lymphocytes packed in here in the mucosa, but there's no capsule. Then, overlying the Peyer's patch, you have a specialized flattened epithelium called follicle-associated epithelium. So, instead of the usual columnar epithelium with the goblet cells and tons of microvilli to increase absorption capacity, these follicle-associated epithelial cells are flattened out. And then, interspersed here and there among these flattened epithelial cells are M cells, or microfold cells. These M cells are able to take up antigens from the lumen of the gut and then transcytose the antigens to the other side of the cell. So they bring antigens from the luminal side of the cell to the basal side, where they have a deep pocket-like structure, which is sort of like a docking station for cells, like dendritic cells. B cells, T cells, macrophages, all of these immune system cells that need to know what's shaking in the GI tract. This is how the immune system finds out what kind of antigens are present for the gut that it needs to start preparing for. And then deep in the center of the Peyer's patch, the B cells have been activated and have differentiated into plasma cells to produce tons of IgA. That central portion of the Peyer's patch with all of those plasma cells is called the germinal center. Now let's talk about lymphatic drainage. The lymph fluid drains the lymph nodes where it's cleaned up before going back into circulation. So let's talk about which lymph nodes, specifically parts of the body, drain to. This is a 4-star topic. Pretty much everybody has at least one question about lymphatic drainage on their test. It's usually disguised as a question about a cancer patient with metastasis to a regional lymph node, and then they ask, where is the lymph node region to which this particular cancer might metastasize? So make sure you know these. So here's our buddy Dr. Lewis, and let's take a look at lymphatic drainage, starting with the extremities. The arm and lateral breast cancers are going to metastasize to the axillary nodes. This is why when we do our clinical breast exams, we palpate the axilla very carefully to make sure there's no axillary lymphadenopathy that may indicate breast cancer. And then going to the lower extremity. The calf and dorsolateral portions of the foot drain to the popliteal region. So if you have a foot infection, you might see an enlargement of your popliteal lymph nodes. Make sure you know that. And then the thighs, they drain to the superficial inguinal nodes. If you have an infection in the thigh, then you're going to have some superficial inguinal lymphadenopathy. We'll come back to the superficial inguinal nodes here in a second. Let's look at the lymphatic drainage of the GI tract, which is a little bit trickier. The stomach and the upper duodenum drain to the celiac nodes. The lower duodenum, jejunum, and ileum drain to the superior mesenteric nodes. So it kind of matches the blood supply. The celiac trunk supplies the stomach, and the SMA supplies most of the small intestines. And then the proximal 2/3 of the colon, from the cecum to the splenic flexure, drain to the superior mesenteric nodes as well. When you get to the distal third of the colon, the descending colon and the sigmoid colon, and the upper part of the rectum, all of that drains to the inferior mesenteric nodes. The lower rectum, the anal canal and above the pectinate line, drains to the internal iliac nodes, along with the urinary bladder, the cervix, and the prostate. Below the pectinate line, there's a transition to squamous epithelium, ectodermal tissue, and this region below the pectinate line drains to the superficial inguinal nodes. So everything that's superficial to the perineum is going to go to the superficial inguinal lymph nodes. But the testes are going to drain to the para-aortic lymph node. Make sure you know that distinction. The testes are embryologically not a superficial structure. They descend from within the body cavity. So the lymphatic flow goes back into the body cavity to the para-aortic lymph nodes, which is where the testes come from. That's definitely high yield to know. But the scrotum, and in women the vulva, are superficial structures. And where do we say those superficial structures drain to? The superficial inguinal nodes. Make sure you know that the right arm and the right half of the head drain to the right lymphatic duct. So if there's an obstruction in the right lymphatic duct, maybe because of a cancer, you might see some non-pitting edema in those regions. And then there's the rest of the body, and it ultimately drains to the thoracic duct. And where does the thoracic duct empty into circulation? At the junction of the left subclavian and the internal jugular vein. The junction of the left subclavian vein and internal jugular vein is where the thoracic duct returns lymph back to circulation. Just as we divided the organs of the immune system into primary lymph organs and secondary lymphoid organs, we can talk about the functions of the immune system into innate immunity and adaptive immunity. The innate immune system is fairly primitive. It's nonspecific. It includes cells like macrophages that pretty much attack any foreign invader. And that's what I mean by nonspecific. We don't have some macrophages that only attack Salmonella and some that only attack MRSA. Macrophages go attack any foreign pathogen. And they are nonspecific and indiscriminate. The innate immune system also responds fairly rapidly. Because it's nonspecific, you don't have to ramp up and activate lymphocytes and produce a bunch of antibodies. That takes time. So the innate immune system responds rapidly. The innate immune system also has no memory. We'll talk more about memory cells and the importance of immune system memory later on, but the reason that vaccination works is that you're sort of teaching the immune system what measles virus or the chicken pox or meningococcus looks like, so that if you come in contact with those pathogens later on, the immune system will remember that and attack and prevent infection. So again, the innate immune system has no memory. So the memory part of the immune system involves the adaptive immune system. These cell types that we're talking about with the innate immune system include macrophages, as I mentioned, which are types of phagocytes that engulf the pathogens and then digest them. Another type of innate immune system cell are the dendritic cells. Dendritic cells are phagocytes that are professional antigen-presenting cells, which means they phagocytose the pathogens, digest them, and then put the little antigenic piece of that pathogen on the outside of the dendritic cell to present that antigen to the helper T cells. The helper T cells then become activated. It's almost like they're flagging down the T cells by waving a banner and shouting, "Hello, I found something you need to attack." But we're going to cover the antigen presentation in detail in the next video. You have neutrophils, which are another type of phagocyte. And also the mast cells, eosinophils and basophils, which are responsible for secreting histamine and cytokines and signaling chemicals that attract other cells to mediate inflammation. You can think of these as being nonspecific signals, and the inflammation is certainly a messy, indiscriminate, nonspecific process. And if you've ever had an allergic reaction, you know that mast cells and basophils can degranulate rapidly and whip up a little histamine. So again, the innate immune system responds rapidly. Another component of the innate immune system are the natural killer cells, or NK cells, which is interesting because NK cells are actually a type of lymphocyte, and otherwise the lymphocytes are a part of the adaptive immune system. But NK cells are considered a part of the innate immune system. The innate immune system also includes complement proteins, which obviously aren't a cell type, but complement proteins can be a bit indiscriminate. We'll talk about complements in another video. And the last part of the innate immune system is just the skin and other barriers, like gut wall mucosa, which are like a wall that try to keep everything out. Very nonspecific and indiscriminate. Then to contrast the innate immune system with the adaptive immune system. The adaptive immune system is less primitive and more advanced. Instead of being non-specific, the adaptive immune system is very specific. It's like the immune system 2.0. You have very specific receptors on the T cells and very specific antibodies on the B cells that allow these cells to respond only to specific antigens. So there's some genetic variation and recombination that allows the receptors and antibodies to become very, very specific to the antigen epitopes they bind. We'll look at that in another video. The adaptive immune system also generates memory, so that you don't get measles or chickenpox every time it starts going around. And as we've said, the adaptive immune system takes a bit longer to get activated the first time as it sees a pathogen, although the second time around, when the memory kicks in, it responds much more quickly. The adaptive immune system consists of T lymphocytes and B lymphocytes, and also the circulating antibodies, which are produced by plasma cells. In what cell type do we say the plasma cells are derived from? From B cells. Plasma cells are just B lymphocytes that have matured into antibody factories. All right, let's go ahead and go through your end of session quiz. Let's get started. First question. To which lymph nodes do the sigmoid colon and rectum drain? The sigmoid colon and upper part of the rectum drains to the inferior mesenteric nodes. The portion of the lower rectum that's above the pectinate line drains to the internal iliac nodes. And then the distal to the pectinate line, the anal canal drains to the superficial inguinal nodes. Next, in what parts of the lymph node would B cells and T cells be located? B cells are found in the follicles of the cortex. T cells are found in the paracortex. Last question. Where would you find Peyer's patches? They're found in the ileum, specifically in the lamina propria and the submucosa of the ileum. That's it for Immunology 1. In the next video, we're going to talk about antigen presentation. I'll see you then.
It's time for another warm-up review. Let's get going. Which muscles and nerves are derived from the second branchial arch? So the mnemonic for the second arch structures is the letter S itself. So you have cranial nerve 7, the facial nerve, and that innervates the muscles of the facial expression. Then the S muscles include the stapedius and the stylohyoid. Next, what is the antidote for each of the following substances? So first we have opioids, so either naloxone or naltrexone. Antidote for benzodiazepines, that's flumazenil. For heparin, the reversal agent is protamine. Warfarin interferes with the liver's ability to make vitamin K, dependent coagulation factors, so the reversal agent is just going to be vitamin K. But since that actually takes several days to really improve coagulation, for immediate reversal of warfarin, we give a prothrombin complex concentrates to replace those coagulation factors that the patient is deficient in. And the last one here is theophylline. You're going to use beta blockers for that. Next, which ECG finding is associated with hyperkalemia? So remember that T waves mirror the potassium level. So with high potassium, you get tall peaked T waves, and with low potassium, you get kind of flattened T waves, maybe also a U wave. All right, that's it for the review. Let's get to that video.
In this video, we're going to discuss the details of antigen presentation. In order for your body system to mount an immune response to anything, the immune system has to recognize it as a threat. For example, a phagocyte, like a dendritic cell, engulfs some pathogen, like bacteria or a virus or whatever, and then digests it. And then the dendritic cell puts a little piece of that pathogen on the surface of the dendritic cell, where the helper T cell can see it and bind to it. So dendritic cells are antigen-presenting cells that are presenting a foreign antigen to a helper T cell. That whole point is to activate the helper T cells, to tell the immune system that there's something, a threat, that they need to start attacking. We'll review the different kinds of T cells and the activation of T cells in another video, but for right now, I just want to go over a couple of really basic points about T cells that apply to the antigen presentation. A T cell precursor begins life in the bone marrow, and then they have to travel to the thymus, where selection takes place, and the cells that are selected to mature into T cells, and the rest that aren't selected, are destroyed. We're going to talk about that later. Remember, they're called T cells because they mature in the thymus, and T is for thymus. Now, a T cell is a type of lymphocyte, but you can't tell a T lymphocyte from a B lymphocyte just by looking at it. So how can you tell them apart? You tell them apart by the different surface markers and receptors on the T cell, and these surface markers are worth knowing for step 1. Every T cell has a very specific T cell receptor, or TCR, that's capable of binding to a very specific antigen. Remember, T cells are a part of the adaptive immune system, which is very specific. So, T cells have a T cell receptor that can bind to a specific antigen. And actually, there are hundreds, maybe even thousands of T cell receptors on the surface of a single T cell, and they're all identical. This T cell might have thousands of receptors for an antigen that's found on the chickenpox, the varicella virus, and this other T cell has thousands of receptors for the measles virus antigen, and so on. Very closely associated with the T cell receptor is a surface marker called CD3. It actually helps the T cell receptor transmit a signal to the T cell when it binds to that specific antigen. So I want you to think about CD3 as a marker for all T cells. Now, just in case you thought this was getting a little too simple, there are different types of T cells, which can also be differentiated by their surface markers. We mentioned helper T cells. Helper T cells have a CD4 surface marker. They're sometimes abbreviated TH, so sometimes they get called T helper cells. Helper T cells or T helper cells, same thing. The other big type of T cells are cytotoxic T cells, which is abbreviated TC. Cytotoxic T cells are identified by a CD8 surface marker. There are also memory T cells and regulatory T cells, which are supposed to be called suppressor T cells, and a few other types that you don't need to worry about for step 1. Thank goodness. Again, the term lymphocyte refers to both T cells and B cells, but around 80% of the lymphocytes in circulation are actually T cells. So when someone talks about lymphocytic infiltration, for the most part, we're talking about T cells. Let's talk about how helper T cells get flagged down by an antigen-presenting cell. Basically, the T cell receptor on a helper T cell will bind to a specific antigen only when that antigen is presented by an antigen-presenting cell. This is because the CD4 marker on a helper T cell is actually very closely associated with that T cell receptor, and the CD4 marker actually links up with a protein complex on the surface of the antigen-presenting cell. This is called the major histocompatibility complex, or MHC, specifically MHC class 2. The way this works is after the antigen-presenting cell phagocytoses that pathogen and digests it, it puts a piece of the antigen on the surface attached to the MHC2 molecule. Then, that antigen-presenting cell can start presenting this antigen to every T cell it meets, looking for one with matching T cell receptors. When it finds one, the CD4 on the T cell has to bind the MHC2 on the antigen-presenting cell. And when that happens, the CD4 and CD3 can start their signaling cascade that ends up activating the helper T cell. And we'll talk about that activated helper T cell in another video. But for now, just remember that CD4 positive helper T cells bind to MHC class II. In the video, when we talk about T cells, we're also going to look at cytotoxic cells. Now, if cyto means cell, what do you think cytotoxic T cell does? It kills the cells. Specifically, cytotoxic T cells are supposed to kill virus-infected cells, or cancer cells. Well, that's all well and good, but how can the cytotoxic T cells recognize the virus-infected cells or cancer cells? How can they tell the difference between a healthy cell and a diseased cell? Well, the answer is the major histocompatibility complex again, but this time it's MHC class I. MHC class I is expressed by basically every nucleated cell in your body. So not the red blood cells, but pretty much every other cell in your body has an MHC class I molecule on their surface. And attached to those MHC class I proteins are peptide antigens that come from inside the cell. Your antigens are slightly different from mine, so those unique antigens get attached to something called the peptide binding groove on MHC class I molecules on the surface of all of your cells. It's kind of like a way for your cells to tell the cytotoxic cells, hey, leave me alone, I'm on your team, I'm one of the good guys. But when a cell is infected by a virus, it starts putting viral peptide antigens in the peptide binding groove of the MHC1 molecules. And that tells the cytotoxic cells, psst, virus, I've got a virus. And the cytotoxic cells are going to recognize that viral antigen and kill the cell. Because remember, a virus-infected cell is just a factory for more viruses, and the only way to deal with it is to kill the infected cell. It's the same kind of thing you do with a cancer cell. Cancer cells start expressing weird cancer antigens in their MHC class I molecules, and that tells the cytotoxic T cells something's up, so they can come attack and kill that cancer cell. So think back. If CD4 was the surface marker for helper T cells, what did we say the marker was for cytotoxic T cells? That was CD8. So just like the CD4 binds MHC class II on antigen-presenting cells, CD8 binds MHC class I on virus-infected cells, or cancer cells, and then the cytotoxic T cells kills those cells. It's pretty easy to keep this straight in your head. You just multiply the CD number by the MHC number, and you get 8. CD4 times MHC2 equals 8, and CD8 and MHC1 also equals 8. How convenient is that? Now let's dig into some genetics of the major histocompatibility complex. Like all proteins, MHCs are derived from very specific genes. The genes that code for MHCs are called HLA genes, human leukocyte antigen genes. And just like your MHC1 molecules are unique to you and different from my MHC1 molecules, your HLA genes are different from mine. Again, MHC1 molecules are found on virtually every nucleated cell in your body, and they are coded by 3 genes: HLA-A, HLA-B, and HLA-C. And then HLA-D codes for MHC2. But we have to break them down into 3 subgroups. Remember, MHC2 molecules are only found on antigen-presenting cells, and the genes that code for MHC2 proteins are HLA-DR, HLA-DP, and HLA-DQ. Now, all of these HLA subtypes can be further classified into specific loci and specific alleles. You might divide HLA-DR into HLA-DR1, or DR2, or DR3, and so on. Or, for HLA-B, you can have HLA-B1, HLA-B2, and so on. That's where HLA-B27 comes from, which should sound somewhat familiar to you. The importance of this is that certain alleles and loci are associated with certain diseases. And not too surprisingly, these are generally autoimmune diseases, where the patient's own immune system is attacking from within. And not all of you will have a question associated with HLA and a certain disease, but a few of you will. And the ones that are important to know are HLA-B27, HLA-DR3, and DR4. As we discussed in rheumatology, HLA-B27 is associated with seronegative spondyloarthropathies, and the mnemonic for these is PAIR. The P is for psoriatic arthritis, the A is for ankylosing spondylitis, and the I is for the arthritis you can see with inflammatory bowel disease. And R is for reactive arthritis, or the older name was Reiter syndrome. And then HLA-DR3 and DR4 are associated with type 1 diabetes, which is caused by autoimmune destruction of the pancreatic beta cells. Cells that express MHC2 on their surface are antigen-presenting cells. And there are a handful of cells that can act as antigen-presenting cells, like macrophages, sometimes certain B cells, but by far the most important type of antigen-presenting cells are the dendritic cells. Let's look at number 4 in your study guide. They're called dendritic cells because they're covered with these membranous extensions that look like dendrites on a neuron. And they've got their feelers out, trying to capture any antigen that may come along. Dendritic cells display both class 1 and class 2 MHC molecules. Class 1 because all nucleated cells display MHC1, and class 2 because that's how they present the antigen to the helper T cells. They also have a surface protein called B7, which can bind to the specific proteins on the T cell and either increase or decrease the signal that is activated when the T cell receptor binds to the antigen that is being presented. And B7 is sometimes called a co-stimulatory molecule. It won't activate the T cell by itself, but it can influence the activation of the T cell when other signals are around. Dendritic cells also have a CD40 surface marker, which allows the T cells to activate the antigen-presenting cells. So everybody is stimulating everybody else. We're going to see CD40 and B7 again in the next video. This is just a brief introduction. But CD40 is found on the dendritic cells, but it's also found on other antigen-presenting cells, like macrophages and B cells. So what do dendritic cells do? They're designed to capture antigen. They capture antigen at one location and then present it to T cells in another location. Once a dendritic cell captures an antigen, it changes its shape. It's no longer a cell with those long dendritic arms. It changes the shape so that it can migrate to the lymph node and say, hey, look what I found! I found some antigen. Out in the periphery, dendritic cells serve as peripheral sentinels. A sentinel is like a guard or a watchman or a sentry. This is sometimes called the immature state or the sentinel state. And in that sentinel state, that antigen capturing state, they can capture antigens by 3 different mechanisms. By phagocytosis, by receptor-mediated endocytosis, where the dendritic cell's surface receptors may recognize an antigen and then endocytose that antigen using clathrin-coated pits. And the third mechanism is penocytosis, which is like endocytosis of extracellular fluid and whatever small particles are floating within it. Kind of like cellular drinking of fluid, dendritic cells can replace their entire cell volume within an hour through penocytosis. So it's kind of like a sieve. It's just taking a bunch of stuff in and looking for any antigen it can find within that fluid. So the cell starts in this immature sentinel state, this antigen capturing state. Then, once it's captured an antigen, it makes a transition to an antigen presenting state, which allows it to travel into the circulation. And in this state, the dendritic cell has maximal capacity to present antigen to the helper T cells because it's increased expression of class 2 MHC and increased expression of those co-stimulatory molecules. So they find an antigen and then they switch this cell that presents very well to the T helper cells. There are also different types of dendritic cells. One you may have heard of is the Langerhans cell. A Langerhans cell is a dendritic cell that's found in the epidermal layers of the skin. Sometimes it's referred to as a cutaneous associated lymphoid tissue, or CALT. So the Langerhans cell is a type of dendritic cell that hangs out in the epidermis, and again, once it finds an antigen, it's going to change shape and migrate to the lymph nodes. And then in the interstitial spaces of your organs, except the brain, you have interstitial dendritic cells. let's briefly discuss a pathology that can arise in the Langerhans cell, which is called Langerhans cell histiocytosis. This is a disease where there is an excessive proliferation of Langerhans cell, these dendritic cells in the epidermis. And the problem is these cells are no good at antigen presentation, so they're not able to do their job. These cells express an S100 tumor marker and also a surface marker called CD1A. And the way you would probably see this on your test is that in electron microscopy, these cells are going to have a Burbeck granule, which look like little tiny tennis rackets within the cell. That's it for antigen presentation, so pause the video and try to answer the end of session quiz. First question. What molecules are expressed on the surface of dendritic cells? We mentioned MHC class 1, MHC class 2, a B7 protein, and CD40. Next, what are 3 cell types that are known for presenting antigens to CD4 T cells? These are dendritic cells, macrophages, and B cells. Next, what characteristic finding would you see on electron microscopy of a dendritic cell and a patient with Langerhans cell histiocytosis? That's a Burbeck granule. Last one. Which MHC class is recognized by helper T cells and by cytotoxic T cells? Helper T cells recognize MHC class 2, and cytotoxic T cells recognize MHC class 1. That's it for the session quiz.
It's time for the 3-question warm-up for Immunology 3. Let's get going. What muscles and nerves are derived from the third branchial arch? So this is the stylopharyngeus muscle, which is innervated by the glossopharyngeal nerve, cranial nerve 9. Next question. What drug category is often used in the treatment of myasthenia gravis? So we use acetylcholinesterase inhibitors like pyridostigmine and physostigmine to treat myasthenia. Next, to which class of antiarrhythmics do each of the following agents belong? So first we have sotalol. Technically it's a beta blocker, but its strongest action is as a potassium channel blocker, so it's classified as a class 3 antiarrhythmic, so don't get fooled by sotalol. Propranolol is a typical beta blocker, and it's class 2. Bertilium is a class 3, and quinidine is a class 1. That's it for the warm-up. Let's get to that lecture.
We've talked about how the antigen-presenting cells, like dendritic cells, capture antigen and then present it to the helper T cells in order to activate those T cells to fight pathogens. So in this video, we're going to look at some deeper details about T cells. So where do T cells come from? Well, T cell precursors originate in the bone marrow, but they mature in the thymus. Remember, the T in T cell is also for thymus. And make sure that you remember that the thymus is derived from the epithelium of the third brachial pouch. The thymus is sort of like finishing school for T cells. It's where the T cells go to develop and differentiate and mature. I want you to think about the thymus as a circle, and then within that circle is another circle. The outer circle is the cortex of the thymus, and the inner circle is the medulla of the thymus. The cortex is where the immature T cell precursors start out, and as they mature, they gradually move toward the medulla. So mature T cells are found in the medulla of the thymus, M for medulla and M for mature T cells. And as they move from the cortex toward the medulla, a very important process called selection is taking place, where you're selecting for T cells that you want to keep and removing the T cells you don't want to keep. So how exactly does this differentiation and selection work? So T cells in the cortex of the thymus are immature and unspecialized, and they have both CD4 and CD8 surface markers. They also have T cell receptors, which can bind to antigens in MHC molecules. And the cortex of the thymus displays a bunch of different antigens on MHC1 and MHC2. It's basically looking for immature T cells that have a T cell receptor that's capable of binding to MHC. Not too strong and not too weak, but just right. If they bind too weakly or too strongly, the thymus is going to let them die. But if the T cell can bind just right, the thymus feeds it a survival signal, which keeps it alive. And then, still in the cortex, the T cell precursors that bind really well to MHC1 are going to lose their CD4 marker, and they're designated to eventually become CD8 cytotoxic T cells. And the T cell precursors that bind really well to MHC2 are going to lose their CD8 marker, and they eventually become CD4 helper T cells. And that entire process of selecting the T cells that are capable of binding to the MHC is called positive selection. And that takes place in the cortex. And then negative selection takes place at the cortical medullary junction, which is the border or junction between the cortex and the medulla. Negative selection is where you're selecting out T cells with receptors that bind to self-antigens. So when your thymus finds a T cell that wants to bind to self-antigens and attack your own body, that T cell is selected out and destroyed by apoptosis, so you don't have an autoimmune phenomenon. Only about 2% of the T cell precursors in the thymus actually survive the positive and negative selection process. So most of them get killed off. But the ones that do survive eventually leave the thymus as its naive T cells. But that's not the whole story. Because once those CD4 helper T cells are activated, they can become either T helper 1 cells or T helper 2 cells, which have different functions. A T helper 1 cell stimulates macrophages, and they stimulate cytotoxic T cells. A T helper 2 cell stimulates plasma cells to make antibodies. So let's look at how this differentiation takes place. When a naive helper T cell leaves the thymus, it has a T helper 0 cell. If IL-12 is around, say during a viral infection, IL-12 will induce those naive T cells to become Th1 cells. Now, why do you want more Th1 cells around during a viral infection? Well, let's talk about which cytokines Th1 cells produce. Th1 cells secrete IL-2. And what does IL-2 do? IL-2 stimulates the proliferation of T cells, including the cytotoxic T cells that destroy virus-infected cells, as well as stimulating proliferation of even more of these Th1 cells. Th1 cells also secrete interferon gamma, which activates macrophages to go eat up the invading bacteria, or whatever it is. And interferon gamma also suppresses the proliferation of Th2 cells. So you need to remember that T helper 1 cells secrete IL-2 and interferon gamma, which will stimulate cytotoxic T cells and macrophages. And there's a third substance to know in association with T helper 1, which is IL-10. IL-10 is an inhibitory interleukin. It inhibits proliferation of T helper 1 cells. But if you go back to that initial naive Th0 cell, what if instead of IL-12, there was an IL-4 around? IL-4 induces Th0 cells to differentiate into T helper 2 cells. So what cytokines do T helper 2 cells make? They make IL-4 and IL-5, which stimulate and proliferate the B cells. They also make IL-10. And what did we just say about IL-10? It inhibits T helper 1 cells. So if you're making Th2 cells, they're going to pump out IL-4, which stimulates the production of more Th2 cells. And they're going to pump out IL-10, which inhibits Th1 cells. So you're either going to get more Th1 cells or more Th2 cells. It doesn't make sense to produce both at the same time. IL-10 also inhibits macrophages. So the Th2 cells are pro-B cell and anti-Th1 cell. Make sure you know those different cytokines and these T cell differentiation pathways. Now, before a helper T cell can differentiate into a Th1 or Th2 cell, it has to be activated. And what does it take to activate one of these naive Th0 cells? It takes an antigen presenting cell to present it some antigen. You don't want T cells to be activated until there's something under attack. So a T cell precursor in the thymus goes through the selection process and ends up as a CD4 positive Th0, a naive helper T cell. And then it migrates to the lymph nodes and just hangs out there until a dendritic cell comes galloping in from the front lines with some antigen to present to the lymphocytes there. And regardless of whether you're a T cell or a B cell, lymphocytes need not one, but two signals to be activated. It's kind of a system of checks and balances to make sure you don't accidentally activate them. The first signal is that antigen of the dendritic cell is presenting, and the second signal is the co-stimulatory signal. On helper T cells, you have a specific T cell receptor plus a CD4 surface marker. That T cell receptor recognizes an antigen that is presented by the MHC2 molecule on the antigen presenting cell. And remember, when we said that antigen presenting cells, like dendritic cells, also have a B7 protein on their surface. That's involved with a second signal, which is called co-stimulatory signal. And remember that B7 is kind of a family name. The specific B7 protein can either be a CD80 or a CD86. But either way, that B7 protein on the APC will bind to a CD28 surface marker on the T cell, and that's going to activate the T helper cells to start producing cytokines. If it's a Th2 cell, it's going to start producing IL-4, IL-5, and IL-10 to stimulate B cells and suppress the Th1 cells. Or, if it's a Th1 cell, then it's activated, it will start producing interferon gamma and IL-2. Now what about the cytotoxic T cells? How do they get activated? Cytotoxic T cells are looking for virus-infected cells to kill, right? Or cancer cells. And these cytotoxic T cells have a T cell receptor, but instead of CD4, they have CD8, and that recognizes TCR-CD8 complex. It recognizes an antigen presented on an MHC1 molecule. So CD8 recognizes MHC1. That's your first signal. The second signal is that, again, the T cell has a CD28 surface marker that recognizes a B7 protein on the antigen presenting cell. And then, once that cytotoxic T cell has been activated, it can go kill all the infected cells. And also, the IL-2 that the Th1 cells are making is going to stimulate clonal expansion of more of these cells with a specific T cell receptor, which allows the body to kill even more infected cells. Now, while we're talking about lymphocyte activation, let's go ahead and talk about how to activate B lymphocytes in order to make antibodies. Here are the four main steps. The first step is that a helper T cell gets activated, and we just covered that. The next step is that the B cell endocytoses the antigen and presents it to the helper T cell. Remember, B cells can behave like antigen-presenting cells, so they're capable of bringing in antigen. The antigen binds to a surface immunoglobulin, and then the B cell swallows it up, digests it, and puts a piece of that antigen on the MHC2 molecule on the B cell surface, so it can present the antigen to the T helper cell. The T cell recognizes that antigen with its T cell receptor. The third step is another co-stimulatory signal. The B cell has a CD40 marker on its surface, which binds to a protein on the T cell called CD40 ligand. And finally, the first three steps have now induced this T helper 2 cell to produce IL-4 and IL-5, which stimulates the proliferation of B cells and immunoglobulin class switching of the B cells. We'll talk more about the functions of the B cells in another video. But in a sense, the B cell engulfs the antigen and then asks the Th2 cell for permission to fight the antigen. That's pretty much it for the helper T cells. They help by secreting lots of cytokines to recruit other cells like macrophages, or they help by promoting B cells to proliferate and make more antibodies. Helper T cells don't actually do any of the killing, but they call in reinforcements and direct the battle. Before we talk about the other kinds of T helper cells, let's do a quick whiteboard review to go over the actions of these cytokines that are produced by and regulate helper T cells one more time. IL-2 stimulates proliferation of T cells. IL-2 also helps T cells differentiate into CD4 helper T cells, or CD8 cytotoxic T cells, or regulatory T cells, which we'll go over shortly. IL-4 stimulates proliferation of B cells. It also stimulates Th0 cells to become Th2 cells. IL-5 stimulates proliferation of B cells. IL-10 is an inhibitory cytokine, so it inhibits Th1 cells and inhibits macrophages. IL-12 is what stimulates differentiation of Th0 cells to become Th1 cells. Interferon gamma activates the macrophages and also inhibits the proliferation of Th2 cells. And then what produces what? Th1 cells produce IL-2 and interferon gamma mainly. They produce other cytokines like IL-3, but the two main ones to know are IL-2 and interferon gamma. For Th2 cells, I want you to know that Th2 cells produce IL-4, IL-5, and IL-10. IL-12 is produced by macrophages and also dendritic cells. Good job on that. And for your prize, I'm going to give you a smiley face made out of stars. So in the thymus, a T-cell precursors are selected based on their T-cell receptors to either become CD4 helper T-cells or CD8 cytotoxic T-cells. And if a helper T-cells are in charge of directing the battle and recruiting cells to come and fight, cytotoxic T-cells are more like the frontline foot soldiers, the ones actually fighting the battle, killing the enemy. Sometimes they're called killer T-cells. Again, these are CD8 T-cells, so they can bind MHC1 on self cells and kill virus-infected cells, or cancer cells, and or potentially non-self human cells from a transplant. So how do these killer T-cells actually kill these enemy cells? By inducing apoptosis through a couple of different mechanisms. One mechanism is that cytotoxic T-cells release granules of preformed proteins into the cells, including things like perforin and granzyme. Think of these like little grenades that damage these cells. Perforin perforates the plasma membrane, and granzyme is a protease that activates apoptosis within the target cell. A second mechanism for cytotoxic T-cells to induce apoptosis is that the T-cell expresses a molecule called Fas ligand on its surface. And this Fas ligand interacts with the Fas receptors on the target cell, which triggers a series of downstream events and ultimately leads to apoptosis of the target cell. Another type of cell I want to mention are the natural killer cells, or NK cells. These are lymphocytes, and they're kind of like a cytotoxic T cells, and that they also kill other cells by using perforin and granzyme to induce apoptosis. But whereas those true T cells are a part of the adaptive immune system with specific receptors against specific antibodies, the NK cells are part of that non-specific innate immune system. It's more primitive immune system. Unlike cytotoxic T cells, NK cells aren't looking for antigens that are presented on an MHC1 molecule. Instead, NK cells induce apoptosis in cells that do not express MHC class I on their surface at all. Remember, all nucleated cells are supposed to be displaying self-antigens on MHC1, and if they don't, the NK cell can come up and kill it. And that's useful because sometimes virus-infected cells and tumor cells will downregulate their expression of MHC1, almost like they're trying to hide from the cytotoxic T cell. But these NK cells are going to hunt them down anyways. NK cell activity is enhanced by IL-12, by IL-2, and also by interferon alpha and interferon beta. Interferons are very important for fighting off viral infections. Not only do they induce natural killer cells, but they also induce nearby cells to inhibit viral protein synthesis. So if a cell is infected by a virus, it's going to start releasing interferon beta and interferon alpha, and those interferons are going to tell everybody nearby, look, there's a virus around. You need to start generating these substances that will inhibit protein synthesis so that you can be protected. NK cells secrete interferon gamma, which activates macrophages, like we saw in T helper 1 cells. And NK cells can be identified by the presence of surface markers CD16 and CD56. I want you to know that CD16 can bind to antibodies down in the constant region, and CD16 is found on the surfaces of NK cells, macrophages, monocytes, and also neutrophils. So if an enemy cell gets coated with IgG antibodies, NK cells and other cells with this CD16 surface marker are able to recognize antibody-coated cells and then kill them. We call this process antibody-dependent cell-mediated cytotoxicity, or ADCC. There are two more types of T cells that I want to mention just in passing because they aren't very high yield for step 1. Memory T cells and regulatory T cells. Memory T cells are T cells that don't go off to battle. They hang out in the spleen and the lymph nodes and kind of go dormant in active state so that if the body encounters the same pathogen a second time, you have a subset of T cells that have already been activated against that pathogen, and they can be recruited to fight off the pathogen much more quickly the second or third time around. Memory T cells can survive a long time. And in the case of memory cytotoxic T cells, this is what allows some vaccines, especially live virus vaccines, to induce lifelong immunity. Because that population of memory T cells can stick around for so long. Other vaccines may require a booster because they only induce memory helper T cells and memory B cells. But we'll go over that when we talk about vaccines in Immunology 5. And then, regulatory T cells are really poorly understood. Regulatory T cells are basically responsible for regulating the immune system by suppressing it. They inhibit B cells from producing antibodies. They inhibit helper T cells and cytotoxic T cells from doing their jobs. They produce IL-10, which is that inhibitory cytokine, and they release anti-inflammatory cytokines. If your regulatory T cells don't work properly and your immune system isn't suppressed when it should be, you're going to develop autoimmunity and atopic diseases, where the immune system overreacts to a benign, harmless antigen. So, one of the picky details that has been known to turn up on step 1 has to do with a regulatory protein called forkhead box protein P3, or FOXP3. FOXP3 is a protein encoded by a gene found on the X chromosome. And this FOXP3 protein is a transcription factor that helps to control the development and function of regulatory T cells. Patients with a mutation in this FOXP3 gene can't suppress their immune system, and they develop a rare but lethal condition called IPEX syndrome. IPEX syndrome stands for immune dysregulation, polyendocrinopathy, like type 1 diabetes and thyroiditis, enteropathy, which leads to severe diarrhea and failure to thrive, and the X is for X-linked. So the classic presentation is a male infant, because it's X-linked, who has severe chronic diarrhea, neonatal diabetes or thyroiditis, and eczema, atopic dermatitis. And that's the immune dysregulation. That's it for the T cells. Now I want you to pause the video and complete the end of session quiz, and we'll go through the answers. First question, which cytokines are produced by Th1 cells, and which cytokines are produced by Th2 cells? The Th1 cells produce IL-2 and interferon gamma, which leads to the stimulation of T cells and macrophages. Th2 cells produce IL-4 and IL-5, which leads to the activation of B cells and antibody production, and also IL-10, which inhibits Th1 cells. Next, which cytokine inhibits Th1 cells and which ones inhibit Th2 cells? IL-10 inhibits Th1 and interferon gamma inhibits Th2. Next, how do cytotoxic T cells kill virus-infected cells and neoplastic cells? One method was by releasing cytotoxic granules of perforin and granzyme. And the other method was by the Fas ligand on the T cell, which binds to the Fas receptor in the target cell, and that induces apoptosis. That's it for this quiz. I'll see you next time.
Hello everyone, it's time for another warm-up. Let's get going. First one here, which cardiac defect is suggested by each of the following types of murmur? So first one we have here is a pansystolic or holosystolic murmur, heard best at the apex and then often radiates to the left axilla. Think of mitral regurgitation. Crescendo, decrescendo systolic murmur, heard best in the second and third right intercostal space close to the sternum. Think of aortic stenosis. Late systolic murmur, usually preceded by a mid-systolic click. That's going to be mitral prolapse. Early diastolic decrescendo murmur heard best along the left side of the sternum. This is going to be a pulmonic regurgitation, or it could be aortic regurgitation. Late diastolic decrescendo murmur heard best along the left side of the sternum, so this would be a tricuspid stenosis. Next, which heart defect is associated with each of the following disorders? So first we have chromosome 22q11 deletion, so this is associated with truncus arteriosus and tetralogy of Fallot. Keep in mind that the question is asking for heart defects, but definitely also know that that 22q11 deletion is well known to be associated with DiGeorge syndrome. Down syndrome, so that's going to be an endocardial cushion defect. Next we have congenital rubella, so this is either a PDA or pulmonary artery stenosis. Turner syndrome, so this is going to be coarctation of the aorta, also a bicuspid aortic valve. And then we have Marfan syndrome, so think of aortic insufficiency. All right, that brings us to the end of the warm-up. Let's get to that lecture now.
We've discussed how the immune system can be divided into innate immune system, which is primitive and nonspecific, and the more specific adaptive immune system. Well, you can also divide the immune system between cell-mediated immunity and humoral immunity. Cell-mediated immunity is basically T cells or macrophages or NK cells, cells that attack pathogens and eliminate them. And then there's humoral immunity, which means antibodies. The word humoral has to do with body fluids, like back in the old days when they thought disease was caused by evil humors and they would treat by bloodletting or diuretics or making the patient sweat, to get those bad fluids or humors out of the body. So humoral immunity means that this is the acellular part of the immune system that's floating around in the body fluids, specifically antibodies or immunoglobulins. Now, where do antibodies come from? Well, when a mommy antibody and a daddy antibody love each other very much, antibodies come from B lymphocytes. So let's talk about B cells and antibodies. Let's start with some B cell basics, like T cells. B cell precursors originate in the bone marrow. But when T cell precursors leave the marrow and mature in the thymus, the B cell mature in the bone marrow. So just like we said T in T cell stands for thymus, let the B in the B cells stand for bone marrow. And then, once these B cells are mature, they migrate to the secondary immune tissue, like the lymph nodes and the spleen and the molt, where they hang out and wait to be activated. Now, what two cytokines did we say Th2 cells produce that stimulate B cell proliferation? IL-4 and IL-5. But when B cells encounter antigen and are activated, they differentiate into plasma cells to produce antibodies. Or the B cell could become a memory B cell, which goes dormant and waits around in the case that same pathogen reappears. But as you age, the number of memory B cells gradually decreases, and that's why some immunizations can wear off as you get older. Like T cells, B cells have very specific surface markers, and there are 5 surface markers that I think you should know. There's CD19, CD20, CD21, IgM, and IgD. We'll talk about the specific functions of different immunoglobulins in a minute. But remember that IgM and IgD are found on the surface of B cells, in addition to CD19, 20, and 21. We also talked about how B cells can act as antigen-presenting cells because they present antigen to the Th2 cells, and then that Th2 cell can start producing IL-4 and IL-5. So if they are presenting antigen to the Th2 cells, that means the B cells must have MHC2 on their surfaces. And they also must have a B7 protein to help activate the T cell. That B7 protein is important for providing the co-stimulatory signal that stimulates the T cell. And then we mentioned that B cells have a CD40 surface marker that is part of the T cell co-stimulatory signal to activate the B cell. So there are signals for the B cells to activate T cells and signals for the T cells to activate B cells. While we're here, let's talk about the surface markers for other cell types. So these are the surface markers we just covered in B cells: CD19, CD20, CD21, IgM, IgD, MHC class II, a B7 protein, and CD40. Then, T cells have CD3 and a T cell receptor, and also CD28, which is what binds to the B7 protein on the antigen-presenting cell. Cytotoxic T cells also have CD8 surface markers, in addition to many others, and that CD8 is what helps it bind to MHC1. And helper T cells have CD4, plus they have CD40 ligand for activating B cells. The B cell presents the antigen to helper T cells and says, "Check out what antigen I found. What do you want me to do with it?" And the activated helper T cell can tell the B cell to start making antibodies. Natural killer cells have CD56, which is the unique marker for NK cells. And they also have CD16, which is what binds the antibodies that coat infected cells and initiate antibody-dependent cell-mediated cytotoxicity. Let's also look at the surface markers for macrophages. We'll talk about macrophages in another video, but macrophages have CD14 marker. And then macrophages are another type of antigen-presenting cells, so they have MHC2 and B7 protein. And they have a CD40 marker, just like the B cells, which the Th1 cells use to make them more sensitive to interferon gamma. Again, we'll talk about macrophages separately. And then, just as a reminder, all nucleated cells have MHC1. Now, let's talk about antibodies or immunoglobulins. There's some very important 4 or 5-star details out there that you really need to know for your exam. Let's start with the structure of antibodies. There are 2 heavy chain regions and 2 smaller light chain regions. And at the very tip of each of these is a hypervariable region. It's on the amino-terminal portion of each of them. One important thing to know is that the antibody is held together by multiple disulfide bonds. Anytime you want to hold a polypeptide together outside the cell, you need a very strong type of bond, like a disulfide bond. Disulfide bonds are some of the strongest bonds around, so just remember that as a generalization. Disulfide bonds keep things together outside of the cell. And it's particularly important to know where these disulfide bonds are found on the antibody. Again, these picky details are unfortunately important. There are disulfide bonds within the heavy chains and the light chains themselves. There are disulfide bonds connecting the heavy chain to the light chain, and there are disulfide bonds connecting 2 heavy chains to one another. So these disulfide bonds are very important to maintaining the structure and function of antibodies. Now, you can talk about antibodies based on their structure, which involves the heavy chain and the light chain. Or you can look at antibodies based on their function. Antibodies have 2 different functional components, the Fab region and the FC region. There's a Fab region, or antigen binding fragment, which includes that variable region on the amino-terminal end of the antibody. AB for antigen binding. This is what determines the idiotype, the unique antigen to which that antibody can bind. This antigen binding pocket can only bind to one very specific antigen. Again, this is a part of the adaptive immune system, which is very specific. And just like the T cell receptors can only bind to a very specific antigen, it's the same is true for antibodies. Plasma cells will express only one particular type of antibody that binds to one particular antigen. And that Fab fragment consists of 2 light chain regions and 2 heavy chain regions. You need all 4 regions to make the Fab fragment. Then, the FC region is the constant region, found on the carboxy terminal of the antibody. It only includes the heavy chains, and the FC region is the part of the antibody that can bind to the complement. So there are lots of C's associated with the FC region. It's constant, it's on the carboxy terminal, and it's complement binding. We also saw earlier that this portion of the antibody is recognized by CD16 on NK cells. And it's the FC portion that determines the isotype of that antibody, whether it's an IgG antibody, or IgM, or IgA, or whatever. We'll talk about the function of those specific isotypes in a little bit. So the Fab portion determines the idiotype, and the FC portion determines the isotype. There are 5 different types of heavy chains: mu, delta, gamma, alpha, and epsilon. and each of these heavy chains corresponds to a specific antibody isotype. If an antibody has a mu chain, and that makes it an IgM antibody. Delta corresponds to IgD, gamma corresponds to IgG, alpha corresponds to IgA, and epsilon corresponding to IgE. Now, there are only 2 different types of light chains, 5 heavy chain regions and 2 types of light chains, lambda and kappa. There's no functional difference between lambda and kappa chains, not in the same way that there's functional difference between heavy chain isotypes. So an IgM antibody with a lambda light chain is really not that much difference from an IgM with a kappa light chain. But in humans, the normal ratio of kappa to lambda is about 2 to 1. For every 2 immunoglobulins with a kappa light chain you have, 1 immunoglobulin has a lambda light chain. So if you're looking at 150 different types of antibodies in a particular human, the odds are 100 of those antibodies will be kappa and 50 will be lambda. Now, how is that useful? Well, consider a case of multiple myeloma, which is a neoplasm of plasma cells, where one plasma cell undergoes malignant transformation and starts replicating itself over and over and over. In multiple myeloma, you have a huge proliferation of an original malignant plasma cell. So all of those malignant cells are producing the exact same antibody with the same Fab fragment and the same light chains. And that means the ratio of lambda and kappa is going to be skewed. Instead of seeing 2 to 1, it might be 50 to 1 or 1 to 50. And then one last detail regarding the structure of antibodies is the terminology used to describe the composition of the antibody in terms of light chains and heavy chains. So, in an IgG antibody, there are 2 gamma regions, which would be your heavy chains. And it would either have to be 2 lambda or 2 kappa light chains, right? So, IgG would have 2 gamma and 2 lambda, or 2 gamma, 2 kappa. So, what do antibodies do? The short answer is that they bind antigens, either soluble antigens that are just floating around in the serum, or they bind to surface antigens on the pathogens, such as antigens on the surfaces of bacteria. This can neutralize the pathogen, making it harder for the bacteria to cause trouble. This can opsonize the pathogen, making it tastier to the macrophages to gobble it up. And antibodies can activate complement, which eventually build membrane attack complexes and kill the bacteria. We'll talk about that in the video on complement. Let's think for just a moment about how the hypervariable Fab region of antibodies are created on a genetic level. DNA is not supposed to vary, right? But with antibodies, we need a way to create variations in the genetic code for the Fab region in order to create antibodies that can bind every possible antigen you can imagine. The process that allows this genetic variability is called VDJ recombination. The VDJ recombination is rearrangements of the specific segments of DNA, which are called the variable V segment, the diversity D segment, and the joining J segment. These segments compose the coding regions for each specific antigen receptor on B and T cells. So this process explains not only antibody variability, but also the variability in antigen specificity we see with T cell receptors. Any protein on a B cell or T cell that's binding to an antigen is going to be created from this VDJ recombination process. The rearrangement process begins at the breaks in the double-stranded DNA at the recombination signal sequences, which is abbreviated RSS. These signal sequences flank each of the V, D, and J coding regions. After the DNA has broken at these specific breakpoints, the VDJ recombination is initiated by a protein called the Recombination Activating Gene Complex, or RAG, RAG1 and RAG2. RAG1 and RAG2 recognize the RSS sequences that flank each of these coding regions. That's going to initiate the recombination process. And then one last important point here is the mutation in either of these RAG genes, and it causes the inability to initiate this VDJ rearrangement and the arrest of a B cell and T cell development. This recombination is fundamental for the adaptive immune system to work. If a B cell can't generate immunoglobulins, it's pretty worthless. So make sure you know the big steps in the recombination process. Now, let's talk about the different immunoglobulin isotypes. We said that IgM and IgD are found on the surface of B cells, but some isotypes are found in the circulating blood. Let's talk about those circulating immunoglobulins first. The main circulating immunoglobulin is IgG. It is the most abundant immunoglobulin in the blood, and the IgG is part of the delayed immune response to an antigen. When you encounter an antigen for the first time, you don't have any plasma cells primed to make the antibodies against that antigen. It takes weeks to develop a population of B cells that make that specific antibody idiotype and convert them to plasma cells and start cranking out that antibody. That's why IgG is so important in the secondary immune response, or delayed immune response. Let's talk about the important functional characteristics of IgG. IgG fixes complement. It crosses the placenta, so this is the immunoglobulin that's going to keep your baby healthy, not only in utero, but also for the several weeks after the birth. IgG opsonizes bacteria and makes them more tasty for macrophages, and IgG binds to the viruses and bacteria toxins and neutralizes them. So basically, IgG can do all of those things we mentioned earlier as the function of antibodies. IgG is sort of your prototypical antibody. Let's move on to IgA antibody. IgA antibody is the primary antibody that's secreted by MALT, the mucosa-associated lymphoid tissue. IgA secreting plasma cells are found in the mucous membrane and along the GI tract. IgA exists as a monomer in circulation, but the secretory IgA, which is secreted by the GI tract in tears and mucus, saliva, breast milk, or colostrum. IgA is a dimer with the constant regions linked end to end, so the Fab fragments are pointing in two different directions. In order to get into the lumen of the gut, secretory IgA has to pass through the epithelial cells by a process known as transcytosis. And while it's passing through, it picks up glycopeptide called secretory that wraps around the IgA, kind of like a suit of armor to protect it from the gastric acid or whatever harsh environment it's heading into. Next, there's IgE, which mediates immunity to parasites, especially parasitic worm infections. It does this by activating eosinophils. You can remember IgE and eosinophils. IgE's other claim to fame is that it can bind to a specific receptor found on mast cells and basophils, and then when that bound IgE binds to an antigen, it starts cross-linking with other IgE antibodies. And that causes the mast cells and basophils to degranulate and start releasing histamine and a bunch of other substances that can lead to type 1 hypersensitivity reactions, or allergic reactions. So for IgE, think about parasites and think about allergies. So those three, IgG, IgA, and IgE, are all the circulating antibodies. And we said that IgM and IgD are surface antibodies. I want to talk about IgM next because it swings both ways. It's found on the surface of B cells, and it's also found in circulation. When it's attached to the surface of B cells, IgM is a monomer, but in circulation, it exists as a pentamer, with 5 IgM antibodies linked together, kind of like spokes on a wheel. IgM is a part of the primary immune response of an antigen. When you encounter an antigen for the first time, the antibodies generated are mostly IgM. IgM is the primary immune response, and IgG is the secondary immune response. So, say a patient comes in with an encephalitis-like illness, and you're worried he has West Nile virus. You're going to check the patient's blood for both IgM and IgG antibodies to West Nile. If the anti-West Nile virus IgM antibody is elevated, it means the patient does in fact have an acute infection. If the IgG antibody is elevated, that means the patient had West Nile virus sometime in the past, but it's not necessarily an acute infection. The other thing you should know about IgM that is different from IgG is that IgM does not cross the placenta, mainly because that pentamer is just so big it can't. And then finally we have IgD. We don't really know too much about IgD, except that it's found on the surface of B cells, and there are also very low levels of IgD found in the serum. So that's it for antibodies. So now let's work through the end of session quiz. Let's go over the answers. First question, which surface markers are found on B cells? The ones that are unique to B cells include CD19, CD20, CD21, IgM, and IgD. Then they also have MHC1 because all nucleated cells express MHC1, and they have MHC2 and a B7 protein, either CD80 or 86, and also CD40. Which immunoglobulin isotype fits each of the following statements? The first one, associated with allergies, binds to mast cells and basophils and causes them to release histamine. That's IgE. The most abundant immunoglobulin in the blood, IgG. IgA is more abundant overall, but IgG is the most abundant immunoglobulin in the blood. Present in large quantities on the surface of many B cells is both IgD and IgM. Crosses the placenta and additionally confers immunity to the neonates. That's IgG. Can occur as a dimer. Secretory IgA. Largely confined to the intravascular pool and is predominant early antibody frequently seen in response to infectious organism with complex antigens. This is IgM. Distributed evenly between the intravascular and extravascular pools is IgG. Predominant immunoglobulin in secretions, that's IgA. And can be a pentamer, that's IgM. Last one. What initiates recombination of VDJ sequences? That's the RAG1 and RAG2 complexes. Remember, RAG stands for Recombination Activating Gene. That's it for this video. We'll see you next time.
It's time for another warm-up. Let's get going. Which organism produces an exotoxin that works via each of the following mechanisms? So first one, we have inactivates EF2. So this one is going to be Corynebacterium diphtheriae, also Pseudomonas exotoxin A. Next, we have activates GS. So this is going to be Vibrio cholera, but also think about that ETEC, heat labile toxin. Disables GI. So this is going to be pertussis toxin. Bacterial adenylate cyclase. This is going to be Bacillus anthracis. Blocks GABA and glycine. This one is Clostridium tetani, causing lockjaw. Next question, what drug is used to help make the diagnosis of myasthenia gravis? Well, the actual answer is edrophonium. Now, the brand name is Tinsulin, so sometimes we call this the Tinsulin test. It's too short-acting to be used to treat myasthenia, but it's used to make the diagnosis. Next, which heart sound is associated with dilated congestive heart failure? So think about the dilated ventricle. It's going to be that S3 heart sound. Now, which heart sound is associated with chronic hypertension and maybe a more stiffened left ventricle? That's going to be an S4 heart sound. All right, that brings us to the end of the warm-up. I'll see you next time.
We've just been talking about B cells and antibodies, and in this video, we're going to talk about two issues that are related to antibodies, including immunization and autoantibodies. We've briefly mentioned passive and active immunity. Passive immunity is where you give someone preformed antibodies that were made outside the body to enhance their immune system for a short period of time. And obviously, this provides immediate immunity. An example of this is IgA in breast milk. The IgA in breast milk provides a certain amount of passive immunity against specific pathogens. Another example is rabies. If you get bitten by a rabid dog or skunk or whatever, you can get a series of rabies immunoglobulin shots so that you don't get infected. But these antibodies have a short half-life, around 21 days. That doesn't mean that all the antibodies are going to be gone in 3 weeks. It just means half of them are going to be gone in 3 weeks. So, for the most part, if you give immunoglobulins to provide passive immunity over an extended period of time, you have to dose it around once a month. Active immunity, on the other hand, actually involves exposure to foreign antigens, either because the patient had a previous infection or maybe they were vaccinated against something. You know how vaccines work. You give a killed virus or an inactivated virus or bacterial antigen, like a polysaccharide from a capsule, and you inject that so that the patient's immune system can activate some B cells and some T cells against it and be ready, just in case they ever come in contact with the active pathogen. And instead of providing immediate immunity, active immunity takes a few weeks to develop. With that first exposure, in addition to ramping up the production of T cells to fight the infection and ramping up B cells and the plasma cells to produce IgM and eventually IgG against the pathogen, you're also generating memory T cells and memory B cells, which can stick around for long periods of time, providing active immunity. So most of the immunizations that we routinely give, such as tetanus, influenza, MMR, and pneumococcus, those are all examples of active immunizations. Now, if you have a patient who is exposed to rabies, we give both the rabies immunoglobulin and the rabies vaccine, both passive and active immunity. Or if someone comes in with a wound that is high risk for tetanus, we sometimes give both tetanus immunoglobulin and the tetanus vaccine at the same time. Let's talk about vaccines in a little more detail. We're going to start with vaccines against viruses. As I mentioned, there are different types of viral vaccines, live vaccines and inactivated or killed vaccines. With a live vaccine, you actually give the patient the infection, but it's a very weak, attenuated form of the infection. You're allowing that weakened virus to infect cells so that those cells can present the viral antigens to cytotoxic T cells. So live vaccines lead to cellular immunity. Those cytotoxic T cells will not only kill off the infected cells, but some of those activated T cells will become memory cytotoxic T cells, which go dormant and wait for the next time that virus shows up. That's what provides long-term, maybe even lifelong, immunity. A student sent us a mnemonic from the most common live attenuated virus vaccines. The mnemonic is, attention, please vaccinate young infants with MMR regularly. Attention is to remember that this mnemonic is for live attenuated virus vaccines. The P in please is for the Sabin polio vaccine, which is the oral polio vaccine. The V is for varicella, also known as chickenpox, and the Y is for yellow fever. The I in infants is for intranasal influenza vaccine, infant intranasal influenza, although this was recently taken off the market. MMR is for MMR, measles, mumps, and rubella, and the R in regularly is for rotavirus. Now, because you are actually giving live virus, generally you don't give live viruses to immunocompromised patients. Or if you have a patient that's around immunocompromised individuals, you might not want to give them the live vaccine either, because you don't want to expose the immunocompromised person that they're around. There are some notable exceptions to this rule. MMR and the varicella vaccine and yellow fever vaccine can be given to HIV patients as long as their CD4 is above 200. Remember, just because the patient has HIV doesn't mean the patient is immunocompromised. Yet, if the CD4 count is above 200, you give the vaccine because the benefits outweigh the risks. Measles cause about 40% mortality rate in HIV patients. So as long as the CD4 count is above 200, give those live vaccines. Those are the live virus vaccines. Now let's talk about inactivated or killed virus vaccines. Here, the virus can't infect the host cell, so there's no cell-mediated immunity. You're just putting antigen into the system so it can be recognized by the antibodies on the surface of B cells, and that will eventually produce antibodies against the virus. So that's humoral immunity. And these non-infectious vaccines can also produce the production of memory helper T cells. But one issue with these inactivated virus vaccines is that in some cases the immunity can wear off, so a booster dose might be needed. So killed virus vaccines include the intramuscular influenza vaccine, hepatitis A vaccine, the rabies vaccine, and the Salk polio vaccine, which is not the oral polio vaccine, but the injected polio vaccine. I also want you to know which vaccines are egg-based vaccines. The influenza vaccine and the yellow fever vaccine both use viruses grown inside of eggs. So if you have a patient with a severe anaphylactic egg allergy, they need to be skin tested before giving one of these egg-based vaccines. The MMR vaccine is also egg-based, but it's such a tiny, tiny amount of egg, you don't really need to worry about it. So those are the viral vaccines. Bacterial vaccines tend to be either vaccines against the bacterial toxins, like the tetanus toxoid, or the polysaccharides on the capsule of strep pneumo or H. flu. Or sometimes we actually give killed bacteria, like Vibrio cholera, or other attenuated bacteria like the typhoid vaccine or the BCG vaccine against tuberculosis. But in all of those cases, because the bacteria aren't actually infecting individual cells, there won't be a cellular cytotoxic T cell response. So bacterial vaccines are generally going to induce a humoral B cell response. One of the other things to consider with bacterial vaccines that use toxins or capsular antigens is whether or not the antigens have a peptide component. Peptide antigens, or protein antigens, are called thymus-dependent antigens. Thymus-dependent antigens are antigens that can be presented on the major histocompatibility complex. MHC molecules present peptides, which means the thymus can present that antigen to T cells during selection. This means that when we are exposed to a pathogen, we have helper T cells that are able to mount an immune response to generate IL-4 and IL-5, and that will activate B cells, and that promotes antibody class switching from IgM to the more abundant IgG. So peptide antigens, or thymus-dependent antigens, induce a better immune response and a better immunologic memory. When thymus-independent antigens are non-peptide antigens, which means they cannot be presented to the T cells in the thymus, it also means there will be no T cell response, and the B cell response is weaker, because again, the helper T cells are important for activating B cells. So with thymus-independent antigens, the immunological memory is weaker, and boosters are commonly needed. One good example of an antigen that can't be presented to T cells is a lipopolysaccharide, which is the endotoxin found on gram-negative bacteria. So sometimes when we design a vaccine against encapsulated bacteria, we don't give the lipopolysaccharide component. We conjugate that lipopolysaccharide into a peptide, which helps induce a longer-lasting immune response. Now we're going to talk about autoantibodies, which is when your immune system turns to the dark side and starts making antibodies against your own normal antigens in your own tissue. In the study guide, there's a huge table listing autoantibodies that are high yield for step 1. This is at least a 4-star topic. So as we go through each of these, I want you to write down the disease associated with the antibody. I've kind of grouped them by organ system, which may help you keep them straight in your mind. The first one is antibodies against IgG. This is also called rheumatoid factor, which is associated with rheumatoid arthritis. The next one is anti-citrullinated protein antibody, ACPA, which is a little more specific for rheumatoid arthritis. ANA, or anti-nuclear antibody, is a classic autoantibody for lupus, but remember, this is fairly nonspecific. Anti-Smith antibodies are also seen in lupus, and they are much more specific for lupus. And anti-double-stranded DNA antibodies, these are especially associated with lupus renal disease. Antihistone antibodies should make you think about drug-induced lupus. About 50% of people with systemic lupus will be positive for antihistone antibodies, but 90% of patients with drug-induced lupus will be positive. And remember, the mnemonic for drugs that typically cause drug-induced lupus is SHIP. You can go back to rheumatology if you need to revisit that one. Anticardiolipin antibodies and antiphospholipid antibodies are both associated primarily with lupus, especially patients with hypercoagulability and thrombus. We'll come back to that in a moment. Anti-centromere antibodies are seen in crest scleroderma. So the C in centromere, C for crest scleroderma. Anti-SCL70 is pretty easy. That's because SCL stands for scleroderma, diffuse scleroderma. The other name for this is anti-DNA topoisomerase. Anti-Jo1 is seen with polymyositis and dermatomyositis. Anti-SSA and anti-SSB. Remember, SS is for Sjogren's syndrome, SSA is anti-Ro, and SSB is anti-LA. There's an antibody called anti-U1-RNP, ribonucleoprotein, which is seen in mixed connective tissue disease. And the next one is from dermatology, which is anti-desmoglein, seen in pemphigus vulgaris. Now, if you have an anti-acetylcholine receptor antibody, what disease is that? That's myasthenia gravis. Then we have a couple of GI diseases. Anti-endomysial or anti-tissue transglutaminase antibodies and also anti-gliadin antibodies are associated with celiac disease, where you have a gluten intolerance, and that leads to blunting of the microvilli on your enterocytes and malabsorption. Anti-mitochondrial antibodies are seen in primary biliary cholangitis, and anti-smooth muscle antibodies are seen in autoimmune hepatitis. In the endocrine system, anti-glutamate decarboxylase antibodies are sometimes seen with type 1 diabetes. Anti-thyrotropin receptor antibodies are seen in Graves' disease. These are antibodies that bind to TSH receptors and stimulate the thyroid gland to start releasing too much thyroid hormone. Anti-thyroid peroxidase, or anti-TPO antibodies, are primarily seen in Hashimoto thyroiditis. There are also anti-thyroglobulin antibodies that are seen in Hashimoto's thyroiditis or Graves' disease. So there's a lot of overlap in the thyroid antibodies. And then a few autoantibodies that we see in certain kidney diseases. Anti-basement membrane antibodies are seen with Goodpasture syndrome, and that can affect both the kidneys and the lungs. C-ANCA is seen with granulomatosis with polyangiitis, or the older name for this was Wegener's granulomatosis. This can also affect both the kidneys and the lungs. So link those two together in your mind. If you see kidney disease plus lung disease, think about Goodpasture syndrome and granulomatosis with polyangiitis. And then there's P-ANCA antibodies, and these are seen with the type of glomerulonephritis called pauci-immune crescentic glomerulonephritis, and also Chug-Strauss syndrome, and a type of vasculitis called microscopic polyangiitis. I want to circle back and talk about antiphospholipid syndrome, which can get a little confusing, so don't get too hung up in the details. Cardiolipin is a component found in the inner mitochondrial membrane, and for whatever reason, individuals with anti-cardiolipin antibodies are prone to thrombosis and excessive blood clotting. There's another substance called beta-2 glycoprotein, which is a glycoprotein that binds to cardiolipin in vivo. And anti-beta-2 glycoprotein antibodies will also cause thrombosis and clotting. Both of these autoantibodies are sometimes lumped together as antiphospholipid antibodies. Or, if you have blood clots and if you start testing positive for this, we might say that you had antiphospholipid syndrome. Since all of these antiphospholipid antibodies are frequently seen in lupus patients, this is sometimes called lupus anticoagulant, because these patients will have a prolonged PTT, partial thromboplastin time. So based on the labs, they look like they are anticoagulated. But again, these antibodies are actually prothrombotic, and they are not anticoagulants at all. Confused yet? Me too. Here's the bottom line. All of these terms, anti-cardiolipin antibodies, anti-beta-2 glycoprotein antibodies, antiphospholipid antibodies, and lupus anticoagulant, they all refer to the same group of antibodies. You don't need to worry about the subtle differences between them for step 1. Just recognize they all go together and they're associated with lupus and other autoimmune diseases. They can cause recurrent arterial and venous thrombosis, even in fairly young patients. They can also lead to spontaneous abortions, like miscarriages, because of thrombosis. and for some reason, they give you a false positive RPR or VDRL, which is your test for syphilis. So again, don't worry about the details, just know the associations and you should be in good shape. Let's finish up by talking briefly about amyloidosis, which is basically where you have accumulated too much of a specific type of protein in the tissues. That could be accumulation of immunoglobulin, which is why we're going to talk about it here, or it could be another type of protein. As that excess protein is deposited into the tissue, it starts to form beta-pleated sheets, and the tissues start getting thicker and thicker and bulkier, and all that bulk can interfere with the normal function of the tissue. So if you had cardiac amyloidosis, that might cause thickening of the heart muscle and CHF, or it might interrupt the conduction system and cause arrhythmias. Or if you had renal amyloidosis, that might cause nephrotic syndrome and renal failure. Or deposition of amyloid in the walls of the blood vessel, that can lead to strokes and heart attacks. So there are a lot of different clinical presentations. Let's look at some different types of proteins that could cause amyloidosis. This isn't very high yield, but it is fair game. There's immunologic amyloidosis, which can be seen in multiple myeloma, which is where you have proliferation of the plasma cells that are making way too much immunoglobulin. That's specifically the antibody light chains that cause problems. They coalesce to form AL proteins. AL is for amyloid light chains. This is also called primary amyloidosis. Then, secondary amyloidosis occurs when you have chronic inflammation, and that leads to an accumulation of an excessive amount of acute phase reactant known as serum amyloid-associated protein. So if you have a chronic inflammatory disease, like rheumatoid arthritis, you have too much of this serum amyloid-associated protein, and that can form these AA protein molecules that then form amyloid. You can have excessive amylin, which is a minor hormone secreted by the pancreas along with insulin. And in the cases of type 2 diabetes, where there is insulin resistance and the pancreas is working overtime to make extra insulin, it's also going to make extra amylin, and that's going to form AIAPP protein, and that's going to cause amyloidosis. If you have a medullary carcinoma of the thyroid, where there's too much calcitonin produced, that excess calcitonin can form ACAL protein, which can cause amyloidosis. In Alzheimer's disease, there's something called amyloid precursor protein, or APP, that is cleaved to form beta amyloid, or A-beta, which builds up in the brain. So in a way, Alzheimer's disease is amyloidosis of the brain. It's not diffuse multi-organ amyloid, just discrete plaques of this A-beta protein in the brain. And there's dialysis-associated amyloidosis, which is where a protein called beta-2 microglobulin builds up. So be aware of these different amyloidoses. Not incredibly high yield, but definitely fair game and possibly worth knowing. One thing that's definitely worth knowing is how to diagnose amyloidosis based on a biopsy. You need to know that you use a Congo red stain, and that will cause the amyloid protein to light up bright green under the polarized light. You might see it described as apple green birefringence. Now, it's time for the end of session quiz. Let's get started. First question. Which viral vaccines are potentially dangerous to give to patients with an egg allergy? That would be influenza and yellow fever. Next question. Which autoimmune disease is associated with anti-dsDNA antibodies? That would be lupus, systemic lupus erythematosus. Which stain is used to identify amyloid microscopically, and how is this appearance described? It's the Congo red stain, and the amyloid protein shows apple green birefringence under polarized light. That's it for now. We'll see you next time.
It's time for the 3-question warm for Immunology 6. Let's get going. What gives rise to the jugular venous A, C, and V wave? So remember, the A wave is atrial contraction, the C wave is ventricular contraction, and the V wave is atrial filling against the closed tricuspid valve. Next question. Which heart murmur is associated with weak pulses? So this is aortic stenosis. Sometimes this is called pulsus parvus et tardus, which is Latin for weak and delayed pulses. Next question. What findings are associated with Ehlers-Danlos syndrome? So depending on the types of collagen that are affected, you can see things like easy bleeding or bruising. You can have hyperextensible skin. You can have joint hypermobility. And because of all this, there might be dislocated joints. That could also be aneurysms like aortic aneurysms or berry aneurysms as well. All right, that's it for the warm-up. Let's get to that lecture now.
In this video, we're going to discuss hypersensitivity and complement, which may not be terribly exciting, but unfortunately, it's very high yield. For instance, hypersensitivity reactions are at least 4 stars, possibly 5 stars. There are 4 basic types of hypersensitivity reactions, which, imaginatively enough, are called 1, 2, 3, and 4. Type 1 hypersensitivity occurs when free antigen in the blood or on the mucosa binds to IgE on the surface of pre-sensitized mast cells and basophils, and that causes those mast cells and basophils to degranulate and release preformed molecules of vasoactive chemicals like histamine and bradykinin. These are vasoactive amines, so they cause vasodilation and increased vascular permeability, which allows fluid to leave the vessels and to get into the tissue, which then leads to local edema. This is called a wheal and flare reaction, or urticaria, which is commonly called hives. And because these vasoactive amines are preformed, this reaction can occur very, very rapidly. The classic example of a type 1 hypersensitivity is anaphylaxis, like an anaphylactic reaction to a bee sting or peanuts or other foods. So technically speaking, if you say someone is allergic to a medication or food, you're talking about an IgE-mediated phenomenon. If they have a reaction to a food or medication, but it isn't IgE-mediated, you really should not call it an allergy. You might say they're sensitive to it, but they're technically not allergic to it. Another example of type 1 hypersensitivity would be atopic disorders, such as asthma, allergic rhinitis, like hay fever, and eczema. Even though eczema does have a T cell component to it, it's still considered a type 1 hypersensitivity. If you want to determine whether a patient has a type 1 hypersensitivity allergic reaction to something, you could do a skin test or a scratch test. This is usually done by allergists. They prick the skin on the back with a needle a bunch of times, and they put common allergens into those holes. And then they watch to see if the patient develops urticaria. Type 1 hypersensitivity occurs fairly rapidly, so urticaria develops within about 15 minutes. Type 2 hypersensitivity occurs when there are antibodies against cellular antigens. Antibodies bind to cellular antigens and activate the immune response. So this might lead to a complement-mediated damage, or the macrophages may phagocytose the cells, or NK cells may attack and destroy antibody-coated cells. But the main thing is to remember that it started with antibodies against cellular antigens. Specific examples of type 2 hypersensitivity would be autoimmune hemolytic anemia, where you make autoantibodies that attack your own red cells. Another example of hemolysis caused by type 2 hypersensitivity would be acute hemolytic transfusion reaction, where the patient has type A blood and you accidentally transfuse type B red cells, and the patient's anti-B antibodies attack the B surface antigens on the donor cells. We'll talk about that more in a moment. There's also erythroblastosis fetalis, where a pregnant woman's IgG antibodies cross the placenta and attack the fetus's red cells. An ITP, immune thrombocytopenia, they're autoantibodies that attack platelets instead of red cells. Another type of anemia caused by a type 2 hypersensitivity is pernicious anemia, where the autoantibodies attack the parietal cells of the stomach, which produce the intrinsic factor. And there are also autoantibodies against the intrinsic factor itself. Without intrinsic factor, the patient can't absorb vitamin B12 in the terminal ileum, and that leads to B12 deficiency, which then causes anemia and peripheral neuropathy. Myasthenia gravis is a disease where there are autoantibodies directed against the acetylcholine receptors of the neuromuscular junction. Another neuromuscular disease caused by autoantibodies against cellular antigens is Guillain-Barré syndrome, in which there are autoantibodies against the Schwann cells, which leads to demyelination of the peripheral nerves. Graves' disease occurs due to antibodies against the TSH receptor. In pemphigus vulgaris, you have antibodies against the desmosomes or protein desmoglein. Bullous pemphigoid is caused by antibodies against the hemidesmosomes. Goodpasture syndrome is where you have antibodies against the glomerular basement membrane. And acute rheumatic fever is a hypersensitivity in which antibodies against group A strep start attacking the host. Let's move on to type 3 hypersensitivity. Type 3 hypersensitivity is when there are antibodies against soluble antigens, not cellular antigens, like in type 2 hypersensitivity, but free-floating antigens that are then dissolved in the blood. These antibodies bind to the soluble antigens and form antibody-antigen complexes that then get deposited into the tissues. So this isn't really antibodies attacking cells that causes the problem. It's the antibody-antigen complex getting into the tissue that causes the problem. And again, these immune complexes evoke an immune response with activation of complement and recruitment of neutrophils and ultimately a lot of tissue damage. Examples of type 3 hypersensitivity reaction include lupus, rheumatoid arthritis, polyarteritis nodosa, and streptococcal glomerulonephritis. Another example is serum sickness. When you hear serum sickness, I want you to think about antibody-mediated drug reaction. In serum sickness, you produce antibodies to foreign antigens, like drugs. Most drugs aren't proteins, but the drug acts as a hapten, which is a molecule that gets around by a carrier protein, and then that hapten protein complex can bind to antibodies and form immune complexes. So again, serum sickness is an antibody-mediated drug reaction. It's called serum sickness because back in the old days, the pre-antibiotic era, they used to have things like scarlet fever or diphtheria that they would treat with horse serum that contained a bunch of antibodies against the toxins. But after a week or so, some of the patients would start making antibodies against antigens in the horse serum. And those antibody-antigen complexes would then deposit in the tissues, causing fever, rash, urticaria, arthralgias, and proteinuria. So again, nowadays we see drug reactions, but they cause a much milder form than this. One last example of a type 3 hypersensitivity is called Arthus reaction. This is a local subacute reaction where you give an intradermal injection of some antigen, like a vaccine, and then the antigen-antibody complexes get deposited locally in the small vessels around the skin, leading to activation of the complement and local edema, and then tissue necrosis. This Arthus reaction usually occurs 4 to 12 hours after the injection. So it's not as rapid as a type 1 hypersensitivity, but it is a lot more rapid than serum sickness, which takes several days to develop. And then there are type 4 hypersensitivity. This is T-cell mediated. There may be antibodies involved, but ultimately it's caused by the T cells. T cells bind to antigens and then release cytokines like IL-2 and interferon gamma, and those cytokines activate macrophages, and that causes tissue damage. The two buzzwords I want you to remember with type 4 hypersensitivity are delayed hypersensitivity and cell-mediated hypersensitivity. Whenever you hear delayed hypersensitivity, I want you to think of a type 4. And it's delayed because of cellular immunity, and cellular immunity takes a while to get ramped up. It's not like type 1, where you have the antibodies lying around ready to go. With type 4, it takes time to present the antigen to T cells, and then they have to make the cytokines, and then the macrophages have to be recruited. It's a delayed process. Think about poison ivy. It doesn't start itching immediately, it takes some time. Or think about the PPD skin test for tuberculosis. When do you read the PPD? 48 hours after it was given. So some examples of type 4 hypersensitivity would include the PPD skin test, which we just mentioned, Hashimoto thyroiditis, which does involve several antibodies, but it's primarily T-cell mediated, multiple sclerosis, graft versus host disease, where you have a bone marrow graft and the T cells from the donor bone marrow end up attacking the host. Also contact dermatitis, which includes things like poison ivy, poison oak, or some types of latex sensitivity. Although latex can be a true IgE-mediated allergy, or it can cause type 4 hypersensitivity. You can also get contact dermatitis from metals or nickel or cobalt. The big clue from contact dermatitis from nickel is that you see it in areas of the skin that are exposed to nickel. So there might be dermatitis on the wrist from a watch band or the ear from earrings. Or in this photo, there's nickel in the button of the patient's jeans, and right where it's rubbing the skin, it's getting contact dermatitis. So it's usually pretty easy to recognize. I mentioned that a certain type of transfusion reaction as being a type 2 hypersensitivity. But transfusion reactions are not all created equal. There are several different types of transfusion reactions, so let's review those. The first one is an urticarial reaction. This is a minor allergic reaction in which the patient has IgE antibodies against soluble antigens in the donor plasma, and it causes some urticaria, hives, and itching. So, which type of hypersensitivity reaction is this? It is IgE mediated, so it's type 1. You treat this by stopping the transfusion and giving Benadryl. This is an allergic reaction, so treat it with an antihistamine like diphenhydramine or Benadryl. And this is the only type of transfusion reaction that is insignificant enough to continue the transfusion with the rest of the bag. If the urticaria resolves with diphenhydramine, go ahead and continue that transfusion. Now, anaphylactic transfusion reactions are also IgE-mediated type 1 hypersensitivity reactions. So this often occurs within minutes of starting that transfusion. With anaphylaxis, the patient can have shock and hypotension, sometimes angioedema and respiratory distress. This is life-threatening. And that's a major difference between urticarial reaction and anaphylactic reaction. Urticarial reactions are fairly common and mild, while anaphylactic reactions are severe and much less common. The mechanism's the same, but the difference is the severity of the reaction. We don't really understand why some of these patients get severe, life-threatening reactions and others basically have some minor hives. But one classic association is that patients with IgA deficiency are at an increased risk for the anaphylactic reaction. They can make anti-IgA antibodies. So the first time these patients receive a blood product, if there are any traces of IgA in the donor blood, the recipient will recognize that donor IgA as a foreign substance. They'll then form IgG and IgE antibodies against the IgA. Now the next time they receive a transfusion, those new anti-IgA antibodies will attack the traces of IgA in the donor blood, and they will get an anaphylactic reaction. This is a well-established mechanism for anaphylactic transfusion reactions. But most of the time, anaphylactic reactions aren't due to IgA deficiency at all. But it is a classic association, so it might show up on your test. And then for treatment, we give epinephrine, IV fluids to bump up that blood pressure, and oxygen. Sometimes you may even need to intubate them. The most common type of transfusion reaction is a non-hemolytic febrile reaction. It was originally thought that this was due to antibodies against the human leukocyte antigens, HLA, on white cells that was found in the donor blood. But now it's thought that this reaction is caused by cytokines that are generated by cells in the blood while it's in storage. When the blood gets transfused, those cytokines are transfused too, and this causes a fever beginning 1 to 6 hours after the transfusion. You might need to update your book on this one. This is not thought to be type 2 hypersensitivity any longer. So again, there's no hemolysis, just fever, chills, and malaise that begin within 1 to 6 hours of the start of the transfusion. And you treat it with acetaminophen, just as any other fever. A much less common reaction is the acute hemolytic transfusion reaction, which is usually due to ABO incompatibility. This one is a type 2 hypersensitivity, where circulating antibodies bind to the cellular antigens on the donor cells. And the main reason for an ABO compatibility is clerical error. The patient was O negative and you gave A positive blood. because of a clerical mistake somewhere along the way. This is an acute reaction, so it has an onset during the transfusion, within 1 hour, not hours or hours later. The reaction is characterized by severe antibody-mediated hemolysis, leading to fever, chills, tachycardia, tachypnea, and hypotension. The treatment here is aggressive supportive care. The last one I want to discuss is called transfusion-related acute lung injury, or TRALI. This occurs in individuals who have some kind of damage to the pulmonary vascular endothelium, and neutrophils are kind of sequestered in the lungs and primed. When you transfuse the blood products and anti-neutrophil antibodies from the donor activate the neutrophils in the recipient, they release the cytokines and reactive oxygen species and proteases, all that cause pulmonary vascular endothelium to become very leaky. The patient then develops acute pulmonary edema, shortness of breath, usually within 1 to 2 hours of starting the transfusion, maybe up to 6. This is pretty rare, but it is also very severe and is the leading cause of transfusion-related death. These patients need to be intubated, mechanically ventilated, and hemodynamically supported if they develop hypotension. Now, let's talk about complement. The complement system is basically a group of plasma proteins that work together to complement the actions of the immune system by opsonizing and killing pathogens and clearing them out of the way. The good news is you don't need to memorize the entire complement pathway for step 1, but I'm going to review a handful of key things that you should know. First of all, you need to know the 3 pathways of complement activation and what stimulates each pathway. The classic pathway is stimulated by an antigen-antibody complex. Remember, one of the ways that type 3 hypersensitivity reactions cause damage is by activating complement. So that's the classic pathway. IgG or IgM antibodies bind to an antigen and then to a C1 complement protein and activate it. There's also an alternative pathway which does not require antibodies. Complement can be activated by molecules on the surface of microbes, or complement can spontaneously activate itself. There's also the lectin pathway. A lectin is a protein that can bind to a sugar. So, in the lectin pathway, a specific lectin protein called mannose-binding lectin binds to the mannose sugar molecule on the surface of the microbe, and that activates the complement cascade. So, definitely know the initial steps for each of these pathways. The second point I want you to know is that C1 is the starting point for the classic pathway. That's the second time I've said it in the last 45 seconds, so it's probably worth remembering. And there's a specific enzyme called C1 inhibitor that inhibits the cleavage of C1 into active components. And don't get confused, because C1 inhibitor also goes by the name C1 esterase inhibitor. The third major point about the complement pathway that you should know is the very last step, which is the formation of the membrane attack complex, or MAC. The MAC is the end product you get from activating complements. It's a complex of several complement proteins that all get inserted into the plasma membrane of the cell one by one, creating a hole in the plasma membrane and then killing the cell. The specific complement proteins that come together to form the MAC are C5b, C6, C7, C8, and C9. Those are also worth knowing as the components of the MAC. Let's walk through a few more details about some of the individual complement proteins. C3b is important for opsonization, which is basically the process of coating pathogens so they're more likely to appeal to the phagocytes. It's kind of like dipping the pathogen into a chocolate fountain. It doesn't matter what the antigen is. When you dip it in the chocolate fountain, those phagocytes are much more likely to eat it. That's particularly important when it comes to destroying encapsulated bacteria. So patients who are deficient in C3 are more susceptible to encapsulated bacterial infections. We also mentioned that IgG antibodies can opsonize pathogens, so C3b and IgG are the 2 most important opsonins. C3a and C5a play roles in anaphylaxis. To be more specific, C3a stimulates mast cells and basophils. And remember what we talked about Mast cells and basophils release histamine, which cause vasodilation, increase vascular permeability, and it allows the fluid to flow through those vessels, which can lower the blood pressure and lead to anaphylactic shock. That vascular permeability also leads to edema and swelling of the larynx and the tongue, which is why the throat feels like it's closing up during anaphylaxis. And C5a is also involved in neutrophil chemotaxis. Let's talk about some diseases in which the complement system doesn't work correctly. If there is a deficiency of a C1 esterase inhibitor, that causes hereditary angioedema. We don't really understand how or why this works, but it seems to have something to do with an elevated bradykinin. Angioedema in general is bradykinin mediated, and somehow deficiency of C1 esterase inhibitor results in elevated bradykinin. And these patients should never take ACE inhibitors because ACE inhibitors also increase bradykinin level and can cause angioedema as a side effect. Deficiency of C3 leads to recurrent pyogenic sinus infections and respiratory tract infections, specifically those encapsulated bacteria like strep pneumo and Haemophilus influenza. These patients also have increased susceptibility to type 3 hypersensitivity, especially glomerulonephritis. Basically, C3 is needed to opsonize and clear out those antibody-antigen complexes that cause type 3 hypersensitivity. So no C3 means more type 3 hypersensitivity. Deficiency of C5b, C6, C7, C8, or C9 means you can't form the membrane attack complex. So deficiency of any of those will predispose to Neisseria bacteremia, both gonococcal infections and meningococcal infections. And there's also deficiency of decay accelerating factor, or DAF, which is also called CD55. DAF is a substance that helps to protect your own cells from complements that have spontaneously activated itself through the alternative pathway. So DAF deficiency leads to red cell lysis and a condition called paroxysmal nocturnal hemoglobinuria, or PNH. Take a look in your study guide and let's talk about PNH for just a moment. PNH occurs in individuals whose red blood cells are deficient in 2 surface molecules that are involved in protecting the cell from destruction by the complement in the MAG. The first is a glycolipid called GPI, or glycosyl phosphatidylinositol, which is a molecule that anchors various cell surface proteins to the plasma membrane, including decay accelerating factor, or CD55. The second surface molecule that's missing in these patients is called MAC inhibitory protein, or CD59, which also prevents the membrane attack complex from destroying cells. So, if you don't have those 2 molecules, CD55 and CD59, you can get complement-mediated damage of the red blood cells. So your red blood cells are being attacked and lysed by complement, which leads to chronic intravascular hemolysis. And then the hemoglobin in the red cells ends up in the urine and causes hemosideruria and red coloring in the urine, which is a classic finding. Another unique finding in PNH is thrombosis, because platelets also get destroyed and they release their prothrombotic components. Now, how do you diagnose PNH? There are 2 tests to be aware of. Historically, we used a test called Ham's test, which is where you put the patient's red cells in acid. And if they lyse at a very low pH, that's a positive Ham's test, which is diagnostic for PNH. A more common, more modern way of making the diagnosis is to do flow cytometry. Flow cytometry allows you to look for the presence of CD55 and CD59 on the surface of a red cell. If you do flow cytometry and you don't see those surface markers on the red cells, that suggests the diagnosis of PNH. So how do we treat it? These patients need transfusions because of the hemolysis. We also anticoagulate them with warfarin because of the risk of thrombosis. And then there's a drug called eculizumab, which is a monoclonal antibody that inhibits complement and prevents it from attacking red blood cells. But eculizumab costs several hundreds of thousands of dollars a year. All right, now we're ready for the end of session quiz. So work through those questions and then we'll talk through the answers. First question, which type of hypersensitivity is responsible for the following clinical problems? Post-streptococcal glomerulonephritis. That's a type 3 hypersensitivity. What about asthma? Asthma is type 1. And rheumatic fever? That's type 2. A TB skin test? That's type 4. Allergies, anaphylaxis, and hay fever? That's type 1. Polyarteritis nodosa? Type 3. Serum sickness? Also type 3. ABO blood type incompatibility? That would be type 2. Poison ivy is type 4. Eczema is type 1. Contact dermatitis is type 4. And Goodpasture syndrome, that's type 2. Now, what about a patient who suffers from recurrent infections? What complement proteins could they be deficient in? This could be any of our membrane attack complex proteins, C5b, C6, C7, C8, or C9. Last one. Which complement is responsible for neutrophil chemotaxis? That is complement C5a. That's it for this video. I'll see you next time.
It's time for another warm-up. Let's get going. Which heart sounds are considered benign if there is no evidence of disease? So think about a split S1, split S2 on inspiration, S3 heart sound in a patient less than 40 years of age, and then an early quiet systolic murmur. Next, what organisms cannot be stained with gram stain? So treponema is too thin to be visualized with gram stain. Rickettsia and chlamydia and legionella are pretty much intracellular, so they don't take gram stain for that reason. Mycoplasma species like Mycoplasma pneumoniae has no cell wall, so they don't gram stain. And then mycobacteria have a high lipid content. Next, well, what is the cause of Duchenne muscular dystrophy? So it's the deletion mutation within the gene for dystrophin, which is the protein that anchors muscle fibers. So without normal functional dystrophin, you get degeneration of the individual muscle fibers. All right, that brings us to the end of the warm-up. Let's get to that lecture now.
In this video, we're going to discuss a few more cell types that make up the immune system, including macrophages and granulocytes, and then we'll also review some different cytokines. But first, let's talk about the spleen, because the spleen is where a lot of macrophages reside and do their thing. In fact, the spleen is a site where lots of immune cells do their thing, such as B cells and T cells. The spleen is covered by a fibrous capsule, but under the capsule you have red pulp and white pulp. Now the red pulp is red because that's where you find red blood cells, and the white pulp is where you find white blood cells, especially lymphocytes. Now, this area on the edge of the white pulp is called the marginal zone because it's on the margin between the red pulp and the white pulp. The marginal zone is where you find antigen-presenting cells, including macrophages, and these macrophages are presenting antigens to the lymphocytes in the white pulp. So the macrophages kind of patrol the red pulp looking for antigens, and when they find antigens, the macrophages scoop them up and bring them to the marginal zone and then present them to the lymphocytes in the white pulp. Then inside the white pulp you have a central artery, and surrounding that artery is a band of T cells called the periarterial lymphatic sheath, or PALs. So you can remember that T cells are found in P structures. They're found in the PALs in the spleen. They're also found in the pericortex in the lymph nodes. Then the white pulp also contains little follicles of B cells. So B cells are found both in the follicles of the spleen and also the follicles of the lymph nodes. There are a couple of important things that happen in the spleen. Macrophages remove damaged red cells and also encapsulated bacteria. The macrophages in the spleen are cleaning up the blood in a manner of speaking. So if you don't have a spleen, you're going to be more susceptible to encapsulated bacteria, which is why it's extra important to immunize asplenic patients against encapsulated bacteria. So remember, the mnemonic for encapsulated bacteria is even some pretty nasty killers have shiny bodies. For E. coli, Strep pneumoniae, Pseudomonas aeruginosa, Neisseria meningitidis, Klebsiella pneumoniae, Haemophilus influenza type B, Salmonella typhi, and Group B strep. Now, of those, which ones do we have vaccines against? Neisseria meningitidis, Strep pneumo, and H. flu type B. And then think about this. Why would a patient be missing his spleen? Well, one scenario would be a sickle cell patient that has auto-infarcted his spleen over the years. Another scenario would be trauma. So you're in a car accident or something and the spleen gets ruptured and it has to be surgically removed. A third scenario would be a disease called hereditary spherocytosis, which is where the red cells are defective and the macrophages in the spleen keep removing them. So you take out the spleen so that the patient can keep circulating all those red cells. Now the other big function of the spleen is to sequester and store platelets, and also a fair number of red cells. So with splenomegaly, you're going to tend to have thrombocytopenia as more platelets get sequestered by that enlarged spleen. Now we're ready to talk about macrophages. Macrophages begin life as monocytes, which are produced in the bone marrow. Monocytes leave the bone marrow and travel through the circulation for around 8 to 12 hours. Then they migrate into tissues to become macrophages and various other types of phagocytes. In the skin and connective tissue, they become dendritic cells, or sometimes we call these histiocytes, those professional antigen-presenting cells. And dendritic cells are really found throughout the body, but they were first identified in the skin. Dendritic cells are like sentinels or sentries that take up antigens and then migrate to the lymph nodes to present those antigens to T cells and then activate the T cells. In the alveoli of the lungs, in the intestine, and also the spleen, monocytes will differentiate into macrophages. Now, macrophages also act as sentinels, kind of patrolling the GI tract and the respiratory tract looking for pathogens and antigens. And macrophages can present antigen, but they don't travel to the lymph nodes to do it. When macrophages encounter pathogens, they stay put and fight. In the liver, the monocytes become Kupffer cells. In the brain, they become microglia. In the bone, they become osteoclasts. In the joints, the monocytes become type A synovial cells, or type A synoviocytes. Type B synoviocytes are similar to fibroblasts, and they make hyaluronic acid, which is an important component of synovial fluid. But type A synoviocytes are similar to macrophages in that they clean up the synovial fluid. So monocytes in the blood can differentiate into any of these cell types, depending on what target tissue they end up in. Let's talk about some details about macrophages and the role they play in a healthy immune system. We said macrophages are sentinels or sentries, so they kind of hang out in a resting state, cleaning up any debris or any worn out cells, and cleaning up wounds and damaged tissues. We saw that when we talked about wound healing. But macrophages can also become activated to perform other tasks. Macrophages have a CD14 surface marker. When that CD14 binds to lipopolysaccharide, or the endotoxin found on gram-negative bacteria, the macrophage becomes activated. It's like the first awareness that the body has that a bacterial invader has been identified. So once the macrophage is activated, it can perform a variety of functions. So one function of macrophages is to secrete acute phase cytokines like IL-1, IL-6, and TNF-alpha that mediate fever and ramp up the immune response. Activated macrophages also secrete large amounts of IL-12, and we said that IL-12 is what stimulates naive T helper 0 cells to become T helper 1 cells. Then another function of activated macrophages is to present antigen to T cells to make sure they stay active and ready to fight. And one interesting thing is that while macrophages are stimulating T helper 1 cells, the T helper 1 cells are also stimulating the macrophages. So T helper 1 cells produce interferon gamma, which further activates macrophages. And that's an important detail that I want you to know. Macrophages can be activated by interferon gamma as well as being activated by bacterial lipopolysaccharide. Macrophages have another surface marker called CD40, which is part of the co-stimulatory signal that T helper 1 cells use to make macrophages more sensitive to interferon gamma. So that CD40 interaction is the same way that T cells stimulate B cells. So we've mentioned two functions of activated macrophages: to secrete cytokines that ramp up the immune system and to present antigen to T cells. But another important function of macrophages is phagocytosis of bacteria. So we mentioned phagocytosis of cellular debris, but macrophages can also phagocytose bacteria. And we talked about how opsonization with IgG or C3b makes things taste better to macrophages, kind of like dipping the bacteria in chocolate sauce. So macrophages are motivated to phagocytose bacteria that have been opsonized, or coated with that IgG or C3b. And then once the bacteria are internalized, the macrophage digests the bacteria in a lysosome by generating oxygen-free radicals using NADPH oxidase. Now, macrophages can also kill invading bacteria without phagocytosing them, using antibody-dependent cell-mediated cytotoxicity, or ADCC. And finally, we've said that macrophages contribute to chronic inflammation by forming granulomas, where several macrophages combine to form multinucleated giant cells that secrete vitamin D. Now let's talk about granulocytes. Granulocytes are white blood cells that release granules of preformed chemicals, like cytokines or chemicals they use to neutralize or kill pathogens. So there are 4 types of granulocytes: mast cells, eosinophils, neutrophils, and basophils. Now the good news is there are just a few high-yield details that you need to know about this for step 1 or level 1. Mast cells are a type of granulocyte that mainly reside in mucosal tissues in the skin. Mast cells contain granules of lots of things, but one of the most important things is histamine. And we've mentioned that histamine causes vasodilation and increased vascular permeability and some bronchoconstriction. So when mast cells degranulate and release all that histamine, that's responsible for type 1 hypersensitivity. You may have heard of a drug called cromolyn that blocks mast cell degranulation. Now this drug isn't really used very much, but it can be inhaled by asthma patients, or it can be used as a nasal spray for allergy patients. But the problem with cromolyn is it has to be used 3 or 4 times a day. Now the main thing you need to know about eosinophils is the differential for a patient with a high eosinophil count in the peripheral blood, which we call eosinophilia. And in your study guide, there's a mnemonic for the causes of eosinophilia, which is Canada P. So C is for collagen vascular disease. This includes things like polyarteritis nodosa or dermatomyositis. The first A is for atopic diseases, allergies, asthma, Churg-Strauss disease, which is associated with asthma. Allergic bronchopulmonary aspergillosis can cause eosinophilia. Remember, allergies are associated with IgE, and you can link IgE and eosinophils in your mind. The N is for neoplasm. Eosinophilia can be seen in several different malignancies. The next A is for adrenal insufficiency, which is also known as Addison disease. The D is drugs, including NSAIDs, penicillins, and cephalosporins. And the third A is for acute interstitial nephritis, sometimes called drug-induced nephritis, sometimes called allergic interstitial nephritis. So you can think of drugs and allergies can cause eosinophilia, and they can also cause allergic interstitial nephritis or drug-induced interstitial nephritis. Here's a kidney biopsy from a patient who started on a new antibiotic, and then a couple of days later, her kidney started shutting down. They had to do a renal biopsy. And on this renal biopsy, you can see all this inflammation in the interstitial areas. So here are the tubules, and then there are tons and tons of inflammatory cells in the interstitium. And you can see all these pretty red eosinophils. So those eosinophils suggest that this was an allergy to the drug that caused this. And you might see peripheral eosinophilia with this. That's what we're talking about here, peripheral eosinophilia, serum eosinophilia. And then the P is for parasites, things like Strongyloides and Ascaris. Loeffler's eosinophilic pneumonitis is associated with eosinophilia, obviously. But parasite infection is an important classic cause of eosinophilia. And there are also a handful of other causes of eosinophilia, things like HIV, hyper-IgE syndrome, and so on. So remember, Canada P. It's a pretty good mnemonic, eh? Now, I'm not going to say very much about neutrophils or basophils, but I do want to remind you that when a patient has a significant bacterial infection or certain types of inflammatory states, they may develop neutrophilia, which means a high number of neutrophils in the blood. And as the bone marrow starts cranking out more and more neutrophils to fight that infection, the marrow starts pushing out immature neutrophils, which are sometimes called band cells. So here you see a normal nucleus with a clear neutral cytoplasm and then that multilobe nucleus. Now these cells in the center are immature forms called metamyelocytes. And then the cell farthest to the left is a band cell. So instead of that multilobe nucleus, you see the nucleus is sort of thick and curved and band-shaped, sort of C-shaped. So when the bone marrow starts pushing lots of these immature band cells into the peripheral blood, that's sometimes called a bandemia, or you'll hear it called a left shift. Now, there are lots of conflicting stories about where that term left shift comes from, but I kind of like to think about the cells progressing from left to right as they become more mature, kind of like you see on this image. And then as the bone marrow pushes out more immature neutrophils in response to the infection or the inflammation, you see that the cells are shifted to the left. So if you see a shift toward immature neutrophils and immature red cells, so band cells and reticulocytes, that's called a leukoerythroblastic reaction. You might see this with severe marrow disease, like cancer in the bone marrow or myelofibrosis, or as a response to anemia. So we talked about increased white cells, like eosinophilia or neutrophilia. Now let's talk about decreased white cells in the blood, which is called leukopenia. Neutropenia means abnormally low neutrophil count, less than 1,500 cells per cubic millimeter. And when the absolute neutrophil count drops below 500, that's when the patient is more susceptible to infections. Now we see neutropenia in response to severe infections like sepsis, and also things like chemotherapy and possibly radiation. And then marrow failure from aplastic anemia can also cause some neutropenia. And aplastic anemia could be due to drugs or toxins. Another potential cause of neutropenia would be lupus. Now if you have a low eosinophil count, less than 30, that's called eosinopenia. That's pretty rare. I'm not sure I've ever said the word eosinopenia before this very moment. But eosinopenia can be caused by excess glucocorticoids, either exogenous steroids or something like Cushing's syndrome. Remember, we said that adrenal insufficiency or Addison's disease can cause eosinophilia, so it follows that excess adrenal steroids could cause some eosinopenia. And then finally, lymphopenia means a low lymphocyte count, less than 1,500 again in adults. Now the obvious thing that causes this is HIV infection. Remember, HIV causes a decline in CD4 positive lymphocytes. And if that CD4 count's below 200, we call that AIDS. Now, certain congenital immunodeficiency syndromes are also associated with lymphopenia, including SCID, severe combined immunodeficiency, and also DeGeorge syndrome. We're going to cover immunodeficiency syndromes in a separate video. And then again, you can see lymphopenia from radiation, from sepsis, and from corticosteroids. Steroids tend to raise the white count overall, and they raise the neutrophil count, but they lower the lymphocyte count and the eosinophil count. And we've mentioned that sepsis and severe infections can lower both the neutrophil count and the lymphocyte count. We usually think of infections causing a high white cell count, but that's not always the case. Some patients may have a low white cell count. Next, let's talk about cytokines and the various signaling chemicals that regulate the immune system and that different cells use to communicate with one another, starting with the interleukins. Now, there's a good mnemonic for interleukins 1 through 5, which is hot T-bone steak. IL-1 mediates fever, so that's the hot. IL-2 stimulates T cells, so that's the T in T-bone. IL-3 stimulates bone marrow, so that's the bone in T-bone. IL-4 stimulates production of IgE and also IgG, so that would be the E in steak. And then IL-5 stimulates IgA production, and that would be the A in steak. And IL-5 also stimulates eosinophils. And real quick, lots of students seem to get confused about IL-4 and IL-5 and which one stimulates eosinophils. So I told you to link IgE and eosinophils in your mind in terms of type 1 hypersensitivity. But IL-4 stimulates IgE production, IL-5 stimulates eosinophils. Now let's group these cytokines by which cells secrete them. And there can be some overlap. T helper 1 cells secrete IL-2, IL-3, and interferon gamma. So IL-2 stimulates proliferation of T cells, both helper T cells and cytotoxic T cells, and also regulatory T cells. So a lot of immunosuppressant drugs work by blocking IL-2 because shutting down T cells is going to shut down the immune system. IL-3 stimulates growth of bone marrow stem cells, a lot like granulocyte macrophage colony stimulating factor, or GM-CSF, which also induces bone marrow growth. So you can link IL-3 and GM-CSF in your head. And we talked about how T helper 1 cells make interferon gamma, which activates macrophages. We're going to come back to the other interferons in just a moment. So again, T helper 1 cells make IL-2, IL-3, and interferon gamma. T helper 2 cells make IL-3, just like T helper 1 cells did, plus they also make IL-4, IL-5, and IL-10. So we just looked at the function of IL-3, but how about IL-4? Back when we talked about T cell differentiation, we said that IL-4 induces T helper 0 cells to become T helper 2 cells. And we also said that T helper 2 cells are generally pro-B cell because the IL-4 and IL-5 they produce promote B cell proliferation. So IL-4 induces T helper 0 to become T helper 2 cells. It stimulates B cell proliferation. And in that hot T-bone steak mnemonic, we saw that IL-4 stimulates production of IgE and IgG production. So one student suggested the mnemonic beg for it. The BEG is for B cell proliferation, IgE, and IgG, and then it's beg for it, like the number 4 for IL-4. And then IL-5 promotes B cell growth and stimulates production of IgA and stimulates eosinophils. So it might help you to associate IL-5 with snot. Think about mucus containing lots of secretory IgA. And then also think about mucus with allergies, and eosinophils are associated with allergies. So again, you can associate IL-5 with mucus. And then we talked about IL-10 as the inhibitory interleukin because it inhibits the production of T helper 1 cells, and it also inhibits macrophages. And speaking of macrophages, let's review the cytokines that macrophages make. Macrophages make the acute phase cytokines, which are IL-1, IL-6, and TNF-alpha. These cytokines are responsible for ramping up the immune system and generating a fever. IL-1 was the hot in the mnemonic, so it mediates fever. That's pretty much it for IL-1. Same thing with IL-6. It's produced by the macrophages, it's an acute phase reactant that causes fever, and it stimulates the production of acute phase proteins, things like ferritin or CRP. And then TNF-alpha makes blood vessels leaky, and that can lead to septic shock. And it's also involved with recruiting leukocytes. And besides those three acute phase reactants, another interleukin produced by macrophages is IL-8. And I want you to know that IL-8 is involved in neutrophil chemotaxis. So it's a major chemotactic factor for neutrophils that draws neutrophils to the site of inflammation. So there are three substances you should think of as neutrophil chemotactic agents. One of them is IL-8. When we talked about complement, we mentioned that the complement protein C5a is important for neutrophil chemotaxis. And then the third one is leukotriene B4. So interleukin 8, C5a, and leukotriene B4. Group those together in your mind as substances that attract neutrophils. And then the last macrophage-derived interleukin is IL-12. IL-12 induces T helper 0 cells to become T helper 1 cells, and it also activates natural killer cells. Now I promised we'd come back to the interferons. Remember, interferons are primarily antiviral cytokines. Virus-infected cells produce interferon alpha and interferon beta, and those cytokines help neighboring cells avoid virus infection by inhibiting cellular protein synthesis, which blocks viral replication, and they encourage the activation of a ribonuclease that degrades viral mRNA. Interferons also activate NK cells, and one more time, interferon gamma stimulates macrophages. And finally, let's go over a few recombinant cytokines, which are drugs that we give to mimic the actions of cytokines. Now, these aren't too high yield, so I'm going to kind of run through them quickly. Aldesleukin is a recombinant IL-2 drug, which induces the immune system by stimulating T cells. And this is used to treat renal cell carcinoma and metastatic melanoma. And we talked about how IL-3 stimulates the bone marrow, which could be helpful following chemotherapy to help the bone marrow recover. Well, we don't give recombinant IL-3 exactly, but we said that IL-3 has the same function as GM-CSF, granulocyte macrophage colony stimulating factor. So there are a couple of drugs that are GM-CSF analogs, filgrastim and sargramostim. So the GRA is for granulocytes and STIM is for stimulating factor. So grastim is the suffix for these granulocyte stimulating factors. There's also a hormone called erythropoietin that stimulates the bone marrow to make red cells. Now, erythropoietin is made by interstitial cells in the kidney. So patients with renal failure will often have anemia because of erythropoietin deficiency. So we can give recombinant erythropoietin to treat that. And there's a hormone called thrombopoietin that stimulates thrombocyte or platelet production. So patients with thrombocytopenia might benefit from thrombopoietin. And there's an IL-11 derivative called oprelvekin that's also used to treat thrombocytopenia. And there are also some recombinant interferons. Recombinant interferon alpha is used to treat genital warts, hepatitis B and C, Kaposi sarcoma, hairy cell leukemia, and melanoma. Interferon beta is used to treat multiple sclerosis. And interferon gamma is used for chronic granulomatous disease, which is where your macrophages and neutrophils and other phagocytes don't have NADPH oxidase. So you're trying to stimulate macrophages by giving interferon gamma. We're going to talk about chronic granulomatous disease in the video on immunodeficiency syndromes. So now it's time for the end of session quiz. So pause the video and answer those questions, then we'll go over the answers together. First question, what cytokines match each of the following statements? So produced by T helper 1 cells is IL-2, IL-3, and interferon gamma. Produced by T helper 2 cells is IL-3, IL-4, IL-5, and IL-10. Promotes growth and differentiation of B cells is IL-4 and IL-5. Promotes growth and activation of eosinophils is IL-5. Remember, that's associated with mucus. Pyrogens secreted by macrophages include IL-1 and IL-6. Enhances synthesis of IgE, and IgE is IL-4. Remember, E and G is in beg for it. Enhances synthesis of IgA is IL-5. Again, think of secretory IgA and mucus. Mediate inflammation, so these are the acute phase cytokines IL-1, IL-6, and TNF-alpha, which are all produced by macrophages. Secreted by T helper 1 cells and activates macrophages, that's interferon gamma. Make sure you know that interferon gamma is the macrophage activating cytokine. Secreted by T helper 2 cells and inhibits macrophage activation is IL-10. Inhibits production of T helper 1 cells is IL-10. Inhibits production of T helper 2 cells is interferon gamma. Released by virus-infected cells, that's interferon alpha and interferon beta. Supports growth and differentiation of bone marrow stem cells, that's IL-3, or that's the bone in hot T-bone steak. And then supports T-cell proliferation, differentiation, and activation, that's IL-2. Remember, that's the T in hot T-bone steak. And the last question, what's the function of macrophages in the spleen? So macrophages remove encapsulated bacteria and they remove dysfunctional red cells. That's it for now. See you next time.
Oh, excuse me. Just trying to surprise my Jerry with his favorite dinner. Fire seems to have gotten away from me. Oh, what is it? Oh, well, I'll give you one hint. What are the important cytokines and what are their actions? Hot T-bone steak. IL-1 stimulates fever. IL-2 stimulates T cells. IL-3 stimulates bone marrow. IL-4 stimulates IgE production. And IL-5 stimulates IgA production. Hot T-bone steak. Or burnt T-bone steak. Oh.
Welcome back. Let's go over the answers to the warm-up review. First question. A pregnant woman comes to the clinic complaining of new dark pigmentation on her face. What's the most likely diagnosis? This is a classic presentation of melasma. Next question. A woman that is breastfeeding develops redness and swelling of her right breast over a period of 24 hours. Examination reveals a warm, fluctuant mass. What is the diagnosis? This is a classic presentation of mastitis, typically caused by Staph aureus. Next question. A 55-year-old man who is a smoker and heavy drinker presents with a new cough and flu-like symptoms. Gram stain shows no organisms. Silver stain of the sputum shows gram-negative rods. What's the diagnosis? This is Legionella pneumophila, causes Legionnaire's disease. Now let's move on to the lecture.
In this video, we're going to talk about transplant rejection, and then we'll go over the immunosuppressants we use to prevent rejection. So there are 4 basic types of transplant rejection: hyperacute rejection, acute rejection, chronic rejection, and then graft versus host disease, which is kind of unique. Now, hyperacute rejection is something you can actually see taking place in the OR. It occurs very rapidly, within minutes, or maybe within hours of transplanting the organ. This is an antibody-mediated type 2 hypersensitivity reaction, where the transplant recipient has preformed antibodies against donor antigens, and the recipient is rejecting that graft immediately. And we do all kinds of screening and matching to make sure this doesn't happen, because this one's pretty preventable. Now, acute rejection takes longer than hyperacute rejection. Acute rejection occurs within weeks, not within minutes or hours, usually within the first 3 months. And this is cell-mediated. So cytotoxic T lymphocytes are recognizing that graft tissue as being foreign. In other words, the transplanted cells have foreign MHC1 molecules, so the T cells start attacking and killing the transplanted tissue. So to prevent this, or to reverse it, you give immunosuppressants, which will suppress that cell-mediated immunity. And one of the most common mechanisms to block this T-cell-mediated rejection is to block IL-2, because as we've discussed before, IL-2 stimulates the proliferation of all types of T cells. And chronic rejection occurs over several months to years. Most transplanted organs will fail eventually due to chronic rejection, but hopefully you can get 15 to 20 years of good function before that happens. Now, chronic rejection seems to be mediated both by T cells and also somewhat by antibodies. But a big part of it is that antigen-presenting cells present donor antigens to helper T cells, which produce interleukins and generate an inflammatory response that leads to vascular damage and fibrosis of the blood vessels and fibrosis of the transplant tissue itself. And unlike acute rejection, chronic rejection is irreversible. Then one last rejection scenario that we see usually with bone marrow transplants is called graft versus host disease. So when you do a bone marrow transplant, you basically kill all the patient's hematopoietic cells with chemo or with radiation, and then you give them new bone marrow from a donor. And this grafted bone marrow is essentially giving the patient a whole new immune system. Well, graft versus host disease occurs when that new transplanted immune system attacks the host as if it recognizes the host as non-self. So it's graft versus host. So the grafted T cells from this new bone marrow proliferate and start attacking the host as foreign. Now this presents with a maculopapular rash on the neck and shoulders, the ears, the hands, and the trunk. In severe cases, it can blister up and look a lot like Stevens-Johnson syndrome. These patients also get hemolysis and jaundice and hepatosplenomegaly and GI involvement with abdominal pain, diarrhea, and nausea and vomiting.
Now let's talk about immunosuppressant drugs, which we use to prevent transplant rejection. Now for each of these drugs, there are two things I want you to make sure you know: the mechanism of action and the unique side effects. Focus on the side effects that are unique. All these drugs will suppress the immune system and make the patient susceptible to infection. So focus on unique side effects as well as the mechanism of action. So let's start with cyclosporin. Cyclosporin's mechanism of action is that it binds to proteins called cyclophilins in the cytosol of T cells. And then the cyclosporin-cyclophilin complex inhibits calcineurin. And since calcineurin activates the transcription of IL-2, this inhibition of calcineurin prevents the production of IL-2. So cyclosporin is ultimately inhibiting IL-2 production by way of calcineurin, and this disrupts T cell differentiation and activation. Now, when is cyclosporin used? Well, it's used to prevent transplant rejection. It's also used to treat certain autoimmune disorders like psoriasis or severe rheumatoid arthritis. Then for side effects, when you suppress cellular immunity, that's going to predispose to viral infections and lymphomas. But the unique thing I want you to remember is that cyclosporin can cause nephrotoxicity. Cyclosporin causes vasoconstriction of the afferent and efferent arterioles in the kidney, and that leads to hypertension and nephrotoxicity as side effects. The next two drugs I want to cover are tacrolimus, which is also called FK506, and there's a similar drug called pimecrolimus. Now, both of these are used topically for eczema, but tacrolimus can also be given systemically as a serious immunosuppressant for transplant rejection. Both of these drugs work by binding to enzymes called FK binding proteins. And then that complex inhibits calcineurin and inhibits the production of IL-2. Remember, shutting down IL-2 is a common theme for a lot of these immunosuppressants. And then, since these work just like cyclosporin, the side effects are a lot like cyclosporin: nephrotoxicity and hypertension. And then when you use tacrolimus systemically, it can also cause some neurotoxicity like headache and paresthesias. There's a similar drug called sirolimus, which is also known as rapamycin, and it binds to an FK binding protein as well. But the downstream effect of sirolimus is slightly different. Take a look at the study guide. Sirolimus binds to this intracellular protein called FKBP12, or FK binding protein 12, and that leads to inhibition of mTOR, which is the mammalian target of rapamycin. And ultimately, this leads to inhibition of T cell proliferation and response to IL-2. So instead of blocking the production of IL-2, sirolimus actually works on the T cells so that they can't respond to IL-2. And then for the side effects, the main thing to know is that sirolimus is not nephrotoxic. Let's talk about azathioprine next, which is a very commonly used immunosuppressant. It's a precursor of another drug you're going to need to know, which is 6-mercaptopurine, or 6-MP. So azathioprine is a prodrug for 6-MP. Both azathioprine and 6-MP interfere with the synthesis of nucleic acids. And obviously, if you can't synthesize nucleic acids, cells won't be able to divide and replicate, and immune cells won't be able to proliferate. Azathioprine is used in transplant patients, especially kidney transplants, and in autoimmune disorders like rheumatoid arthritis. It's also used off-label for a bunch of things like Crohn's disease and lupus nephritis and all sorts of different things. Then 6-MP is a cancer drug, so we're going to talk more about 6-MP in oncology. But azathioprine, because it's metabolized to 6-MP, it's going to cause bone marrow suppression because the patient can't grow rapidly dividing cells like bone marrow cells. And 6-MP, which is the active metabolite, is ultimately metabolized by the enzyme xanthine oxidase. And make sure you know that enzyme. Xanthine oxidase metabolizes both 6-MP and azathioprine. So the toxic effects of azathioprine can be increased if you're taking the gout drug allopurinol, which inhibits xanthine oxidase. Another immunosuppressant you should know is mycophenolate. Take a look in your study guide. Mycophenolate works by inhibiting inosine monophosphate dehydrogenase, and as a result, it prevents the synthesis of the nucleoside guanine. So inosine monophosphate dehydrogenase is essential in the production of guanine. If you inhibit that, you can't make guanine, and obviously you can't have cell replication. So mycophenolate prevents the rapid proliferation of B cells and T cells. It's used in transplant patients and also off-label for lupus nephritis. And the side effects aren't terribly important, but it can cause hyperglycemia and hyperlipidemia, as well as increased risk of lymphoma and infections. The next item in the study guide is about thalidomide. And we mentioned thalidomide in foundations when we were talking about teratogens. We said thalidomide was a sedative that was given to pregnant women 50 or 60 years ago, but they stopped using it because it causes phocomelia, that severe hypoplasia of the limbs. Thalidomide is one of those drugs with several mechanisms of action, but the reason we're talking about it here is that it suppresses TNF-alpha production. It increases numbers of NK cells, and it also increases IL-2 production. So in some ways, it works like an immunosuppressant. In other ways, it actually stimulates the immune system. But this suppression of TNF-alpha is useful for treating a form of leprosy, or Hansen disease, called erythema nodosum leprosum. And the enhancement of NK cell activity is useful for treating multiple myeloma, because you're stimulating the NK cells to kill the cancer cells. Now, speaking of drugs that block the effects of TNF-alpha, let's talk about infliximab and adalimumab, and then we'll cover a few more designer antibodies that we use as drugs. Infliximab and adalimumab are monoclonal antibodies that bind to TNF-alpha and take it out of commission. These drugs are particularly useful for seronegative spondyloarthropathies, psoriatic arthritis, ankylosing spondylitis, inflammatory bowel disease arthritis, and reactive arthritis, or Reiter syndrome. You can also sometimes use these drugs for rheumatoid arthritis. And then there was one anti-TNF-alpha drug that was not a monoclonal antibody. Remember what that was? Etanercept. Etanercept is a protein that mimics the TNF-alpha receptor, so it binds up all the TNF-alpha. It's called a decoy receptor. Etanercept, decoy receptor. Rituximab is a monoclonal antibody that binds to CD20, which we said was a B cell marker. Remember, B cells have CD19, CD20, and CD21. So rituximab binds to CD20 on the surface of B cells, and that induces complement to lyse the B cell. So rituximab can be used to treat non-Hodgkin lymphoma because most lymphomas are B cell tumors. There's a drug called omalizumab, which is an antibody against IgE. This can be used in severe asthma patients who aren't responding to standard treatment. But it's super expensive, so it's not really used much. Other therapeutic antibodies include digoxin immune fragments, which are actually the FAB portion of an antibody directed against digoxin. So if you have a patient with digoxin overdose that's having severe cardiotoxicity, you can give those immune fragments to bind up all the digoxin. There's abciximab, which is an antibody against the glycoprotein 2B3A receptors on platelets, which inhibits platelet activity. Now, this is used in patients that have recently had an angioplasty and stenting of their cardiac vessels, and also in patients with unstable angina or a myocardial infarction. And trastuzumab is an antibody against a protein called ERBB2 that we use to treat breast cancers that express HER2/neu. Now, for the end of session quiz, we just have one question with a whole bunch of parts. So answer that question, and we'll go over the answers together. All right, let's go over this question. Which immunosuppressant matches each of the following statements? So the precursor of 6-mercaptopurine is azathioprine, inhibits IMP dehydrogenase, that's mycophenolate, binds to cyclophilins and inhibits calcineurin, resulting in the loss of IL-2 production, that's cyclosporin, binds FK binding protein, leading to the loss of IL-2 production, it's tacrolimus or FK506, and also pimecrolimus, binds to FKBP12, leading to inhibition of mTOR and ultimately inhibition of T cell proliferation, that's sirolimus. Used for lupus nephritis is azathioprine, and also mycophenolate and cyclosporin, off-label. And the last one, metabolized by xanthine oxidase, therefore allopurinol will increase its toxicity, that's azathioprine. And that's it for now. I'll see you next time.
Now, here's a little obscure Irish tune called The Ballad of Mike O'Fenelate. What malignancy is associated with mycophenolate mofetil lymphoma? Oh, lymphoma. Mycophenolate increases the risk of lymphoma. And why is mycophenolate mofetil not used in pregnancy? Congenital malformations of the ear and face, and first trimester miscarriage. Mycophenolate mofetil is teratogen. Hey, where are you going? Come back. Oh, I can do requests. Man, I knew I should have played Stairway.
Welcome back. Let's go through the answers to your warm-up review. First question. What is the ECG axis given the QRS deflections in each of the following scenarios? Positive in lead 1, positive in lead 2 is a normal axis. Negative in lead 1, positive in lead 3 is right axis deviation. Positive in lead 1, negative in AVR is normal axis. Next question. Which antiarrhythmic has a side effect of synchonism? Quinidine. And the last question, what stain is required for each of the following organisms or substances? For Cryptococcus, use India ink. For PCP, you use silver stain. And for chlamydia, you use Giemsa stain. Now let's move on to the lecture.
In this video, we're going to discuss immunodeficiency diseases, which are a 5-star topic for Step 1. You should expect to see at least 2 or 3 questions on these. I'm going to highlight the high-yield details, the things that you really need to know so you don't get bogged down in the unnecessary details. In fact, everything you need to know about these diseases for Step 1 is found in your study guide. The first group of disorders we're going to talk about are the T-cell disorders, starting with thymic aplasia, or DeGeorge syndrome. Thymic aplasia is one of the high-yield immunodeficiencies for Step 1. DeGeorge syndrome occurs when the 3rd and 4th pharyngeal pouch fail to develop, which means you have no thymus and no parathyroid glands. Because there's no thymus, the T cells can't mature. And that's why this is a T cell deficiency. And because there are no parathyroid glands, there's no parathyroid hormone, which results in hypocalcemia and tetany. What physical exam signs might be present in a patient with hypocalcemia? There's Chopstick sign and Trousseau sign. Chopstick sign is when you tap the patient's cheek and that causes a facial muscle spasm. You can remember CH for cheek and CH for chopstick. And Trousseau sign is where you tighten the blood pressure cuff on the arm for a minute or so, and that leads to spasm of the arm, called carpal pedal spasm. So remember, T for Trousseau and T for tighten that cuff. Both of these are signs for hypocalcemia. But because there are no mature T cells, these patients have recurrent viral, fungal, and protozoal infections. DeGeorge syndrome is also associated with congenital defects of the heart and congenital defects of the great vessels. 90% of these patients have a chromosome 22q11 deletion. So most of these DeGeorge patients have this chromosomal deletion, but not all of them. You can detect this chromosome deletion with fluorescent in situ hybridization, or FISH. We'll discuss the FISH test in biochemistry. There are a few other T-cell deficiencies, which are generally much lower yield. There's a disease called chronic mucocutaneous candidiasis, which is a disease of the T-cell dysfunction that specifically causes problems with Candida albicans infections. We treat those patients with ketoconazole so they don't get Candida. There's another T-cell disorder called IL-12 receptor deficiency. IL-12 is produced by macrophages, and it induces Th0 cells to differentiate into T helper 1 cells. So if the naive T helper 0 cells are missing that IL-12 receptor, the T cells won't differentiate correctly, and that leads to increased susceptibility to mycobacterial and fungal infections. Next, let's discuss some B cell deficiency disorders, starting with Bruton aglobulinemia. This is an X-linked disease, so it's seen mainly in boys. You can use the B in Bruton's to remind you that this is a B cell deficiency seen in boys. It's caused by a mutation in tyrosine kinase gene, and it leads to low levels of immunoglobulin isotypes. This also causes recurrent bacterial infections after 3 to 6 months of age, because the first few months they're protected by mom's IgG antibodies. So remember, B for Bruton's, B for boys, B for B cells, and B for bacterial infections. In Bruton's agammaglobulinemia, you have low levels of all immunoglobulins, but some patients have a selective immunoglobulin deficiency in which there's deficiency of a specific immunoglobulin isotype. And the ones you're most likely to be tested on are selective IgA deficiency. Most of these patients appear fairly healthy enough that they aren't really aware that there's a major immunodeficiency. All they know is that they have recurrent sinus infections and lung infections. Remember, secretory IgA is found in the mucus, and it's important for protecting the respiratory tract from invading pathogens. So, if they give you a young adult who gets sinusitis 3 or 4 times a year, has had several bouts of pneumonia, start thinking about IgA deficiency. Selective IgA deficiency affects about 1 out of every 600 people in the European descent. It's associated with atopy and asthma. So these patients are more likely to have allergies, asthma, and eczema. But there's not much you can do to treat it, other than having a lower threshold for prescribing antibiotics. It is important to diagnose IgA deficiency because these patients can potentially have an anaphylaxis reaction to blood transfusion and blood products. So if you have a patient with this diagnosis who needs a blood transfusion, make sure you pre-treat them to prevent anaphylactic reaction. Let's discuss immunodeficiency disorders that affect both B cells and T cells. One important one is called SCID, or severe combined immunodeficiency. That should give you a hint that it's combined B cells and T cell deficiency. It's caused by a defect in the early stem cell differentiation, and there are at least 7 different gene defects that can lead to this. But the main defect worth knowing for step 1 is the adenosine deaminase deficiency. We'll see that again in biochemistry when we review the purine salvage pathway. Patients with SCID are deficient in both B cells and T cells, which mean there's a last line of defense as the natural killer cells. SCID patients present with a classic triad that includes severe recurrent infections, specifically chronic mucocutaneous candidiasis, fatal or recurrent viral infections like RSV, varicella zoster, herpes simplex, measles, flu, or parainfluenza, and also pneumocystis pneumonia, or PCP. The second part of the triad is chronic diarrhea, and the third part is failure to thrive. So, know that triad: recurrent infections, chronic diarrhea, and failure to thrive. Another good clue for the diagnosis of SCID is the absence of thymus. The newborn chest x-ray will show no thymic shadow, and that's a good clue. In newborns, the thymus is pretty large because they have a lot of developing T cells. Another immunodeficiency in which you wouldn't see a thymic shadow is DeGeorge syndrome, and that's because they have thymic aplasia and the thymus never develops. Another combined B and T cell deficiency is ataxia telangiectasia, which is another one you'll need to be familiar with. This one has several unique features. Ataxia telangiectasia is associated with a deficiency of IgA. So remember, A for ataxia, A for IgA. But in addition to the IgA deficiency, there's also a problem with T cells and cellular immunity, leading to sinus and lung infections, just like with selective IgA deficiency. This is called ataxia telangiectasia because these patients exhibit cerebellar ataxia. They also have poor, smooth pursuit of moving targets with their eyes. If there is an object moving across the visual field, the patient will have a hard time following it with their eyes, and that's called poor smooth pursuit. These patients also have telangiectasias, specifically telangiectasias on the face, but not until after the age of 5. Ataxia-telangiectasia is a disorder of the defective DNA repair, so these patients are sensitive to radiation. Radiation damages DNA, and then they have a hard time repairing it, so if possible, they should avoid X-rays. They also have an increased risk of lymphoma and acute leukemias. So if you see a toddler with ataxia or poor smooth pursuit, you'd really like to make an accurate diagnosis so that you can monitor for the development of leukemia. AFP is usually elevated in these kids after about 8 months of age, and that can be useful for screening. In that toddler with poor smooth pursuit, you might check an AFP level. So remember, A for ataxia, A for IgA deficiency, and A for AFP. A student sent us a mnemonic for the features of ataxia-telangiectasia, which is simply in the word ataxia. The A and the T are for ataxia and telangiectasia. You can also let the T stand for tracking eye difficulties or poor smooth pursuit. The other A is for acute leukemia and lymphoma, X is for X-ray sensitivity, and I is for IgA deficiency. The final A is for AFP. Another combined B and T cell deficiency is called Wiskott-Aldrich syndrome, and the mnemonic for this one is the word waiter. W and A are for Wiskott-Aldrich, I for immunodeficiency, T for thrombocytopenia and purpura, E for eczema, and R for recurrent pyogenic infections. Wiskott-Aldrich is another X-linked disorder, so this will affect boys, and these boys don't have any IgM against bacterial capsular polysaccharides. They have low IgM levels and high IgA levels. And the last one is hyper-IgN syndrome, which is one of the lower yield topics. With hyper-IgN syndrome, you have too much production of IgM, but decreased production of the other antibody isotypes. But stop and think. If this is a problem with immunoglobulins, wouldn't that actually make it a B-cell disorder? Actually, there are several different genetic defects that result in this phenotype and different inheritance patterns. There's an autosomal recessive variety where the B cells lack the CD40 surface marker. And remember that CD40 binds the CD40 ligand on the helper T cells in order to promote immunoglobulin class switching. So if there's no CD40, there's less class switching, and basically all you get is lots and lots of IgM. But the more common variety of hyper-IgM syndrome is an X-linked variety in which T cells are missing the CD40 ligand, and that's why it's a B cell and T cell dysfunction. Finally, let's review a few immunodeficiency disorders that affect the phagocytes, especially neutrophils. The first one is chronic granulomatous disease, which is definitely high yield. There are several different genetic defects that can cause this phenotype, but the most common ones are X-linked mutation. The underlying problem is a lack of NADPH oxidase, which is the enzyme that generates superoxidase-free radicals. Phagocytes are supposed to engulf the pathogen and debris and then destroy them, but because they lack NADPH oxidase, the phagocytes can't generate free radicals, so they can't kill microbes that are catalase positive. That means that these patients are especially susceptible to infections with Staph aureus and Aspergillus. So, make sure you know that the enzyme that's deficient is NADPH oxidase, and that there are two pathogens, Staph aureus and aspergillosis. Then, to treat chronic granulomatous disease, we give prophylactic Bactrim and prophylactic itraconazole. Interferon gamma is also helpful in the treatment, though the exact mechanism isn't understood. Next is Shiraki-Higashi syndrome. This is caused by a defect in the LYST gene, the lysosomal transport gene, which is important for transporting enzymes into the lysosomes. If you can't get enzymes into the lysosomes, the phagocyte lysosomes are defective. They won't work. So the neutrophils can engulf things, but they can't digest them. So inside the neutrophils, you'll see giant cytoplasmic granules, big lysosomes filled with stuff, but they can't be digested and they can't be destroyed. You need to be able to recognize the classic triad of Shiraki-Higashi syndrome, which is partial albinism, recurrent respiratory tract and skin infections, and a variety of neurological disorders. Everything from peripheral neuropathy to ataxia to seizures. A student sent us this quick mnemonic for the triad of Shirakashi syndrome, which is called the pain triangle. PA is for partial albinism, I for infections, N for neurological disorders. And then you draw some dots inside the triangle to remind you that the cytoplasmic granules that accumulate inside the cells. Let's also briefly mention a very rare disorder called hyper-IgE syndrome. The older name for this is Job syndrome, like Job from the Bible. Hyper-IgE is probably an unfortunate name because while IgE levels are high, that's not really the main cause of the immunodeficiency. Hyper-IgE syndrome is caused by a mutation in the gene for the signaling protein called STAT3, which is part of the Janus kinase signaling pathway, or JAK-STAT pathway. This leads to two main immune system problems: impaired differentiation of T helper 17 cells and the impaired recruitment of neutrophils. We haven't talked about T helper 17 cells because they aren't very well understood, and because they aren't high yield for step 1. But T helper 17 cells do live in the mucosa and produce cytokines that recruit neutrophils to fight opportunistic infections, like staph and Candida. So, some review books classify hyper-IgE syndrome as a T-cell disorder, but I think it's useful to group it with the phagocyte and neutrophilic disorders. These patients also have high levels of IgE and eosinophils, but we don't really understand why. The clinical triad for hyper-IgE syndrome is eczema. You can remember E for eczema and E for IgE. You also have recurrent cold abscesses, or staph abscesses. This disease was first called Job syndrome because these patients were covered with boils, like Job from the Bible. And why are these cold abscesses? You have walled-off area of infection, but the neutrophils can't get in there, so it's not an appropriate inflammatory response. So they aren't hot abscesses, they're cold abscesses. These patients also have coarse facial features, such as a broad nose and a prominent forehead, which is called frontal bossing, deep-set eyes, and thick, doughy skin. These patients can also retain their primary teeth, so they have 2 rows of teeth on top and 2 rows of teeth on the bottom. If you see that, think hyper-IgE syndrome. One last disorder is called the leukocyte adhesion deficiency syndrome. The main thing I want you to know about this one is you have abnormal integrins. Integrins were the molecules that help leukocytes exit circulation and integrate into the interstitium. But with abnormal integrins, phagocytes can't leave the circulation. And one of the ways this presents, in addition to recurrent bacterial infections, is delayed separation of the umbilical cord, which is basically a manifestation of impaired wound healing. So if they give you an infant where the umbilical hasn't fallen off yet and it's been a couple of months, start thinking about leukocyte adhesion deficiency syndrome. That's it for now. Let's do the end-of-session quiz. First question. What are the X-linked immunodeficiencies? We mentioned Bruton A-gamma-globulinemia, chronic granulomatous disease, not always, but most of the time, Wiskott-Aldrich syndrome, and most cases of SCID are X-linked, and most cases of hyper-IgM syndrome are also X-linked. What are the main clinical features of Wiskott-Aldrich syndrome? Remember, the mnemonic for Wiskott-Aldrich is waiter. The first 2 letters, W and A, are for Wiskott-Aldrich, and then I for immunodeficiency, T for thrombocytopenia and purpura, E for eczema, and R for recurrent pyogenic infections. And these individuals have a low IgM and a high IgA. Next, what is the cause of chronic granulomatous disease? And what infections are these individuals susceptible to? The cause of chronic granulomatous disease is the absence of NADPH oxidase. So your phagocytes can't generate oxygen-free radicals. So that makes these individuals susceptible to catalase-positive pathogens, especially Staph aureus and Aspergillus. Last question. A young child presents with tetany from hypocalcemia and candidiasis resulting from immunosuppression. What cell type is deficient in this patient? This is DeGeorge syndrome, which is a T-cell deficiency from thymic aplasia. And then you get the hypocalcemia, which is due to the parathyroid gland aplasia. Then we have a couple of rapid-fire facts. eczema, recurrent cold abscesses, and high serum IgE. That's hyper IgE syndrome, or Job syndrome. And a large phagosomal vesicle in a neutrophil, that's Sheta-Kagashi. All right, that's it for now. I'll see you next time.